A very good morning, all of you. I and Manika, one of the conveners of FFIS 2024, welcome all the members and participants who have joined us on the panel. Welcome to FloraFauna Innovation Summit 2024. It is going to be a day full of learning and experience sharing. We'll listen to our experienced and seasoned professionals and also to the budding innovators and researchers. All in all, we have a platform for everyone, and from each other, we learn and grow. We have Professor V.K. Pathak, Vice Chancellor CSJMU, as our chief guest for today's forum, and Dr. D.B. Ananthanarayana, former Director, Regulatory Affairs, Food and HPC, Unilever Research, as our distinguished speaker. We welcome you both in the forum, sirs. We have Professor S.P.S. Khanuja, former Chairman, FloraFauna Science Foundation, here with us. We also have the esteemed presence of Dr. A.K. Shashne, Honorable Director, CSIR NBRI, will be joining him soon. Professor S.K. Avasthi, Pro Vice Chancellor, CSJMU. He'll also be joining us. Dr. Shantanu Kumar Dubey, Director, ICR Atari. Dr. A.K. Singh, Vice President, FloraFauna Science Foundation. Dr. Neeraj Jain, General Secretary, FFSF. We have Dr. Shilpa Kayastha, CSJMU, and many other distinguished members at the forum. I welcome you all here. We also have Dr. Soni Gupta, convener from CSJMU. Welcome, Dr. Gupta. So without further ado, I would like to invite Professor Sumanpreet Singh Sanuja for initiating the session and his theme lecture on Fasal se Safal. So stage is all yours, sir.
Thank you, Manika. सभी को मेरा नव प्रणाम इस इनोवेशन समिट या नवाचार शिखर सम्मेलन में फ्लोराफोना साइंस फाउंडेशन के इस मंच पर, जो कि पूरा साल आपको विशेषज्ञों के साथ मिलाता रहता है, आज के दिन फरवरी के दूसरे हफ्ते के शनिवार को हमारा ये शिखर सम्मेलन होता है, जहां हम अपने थीम को एक रूप दे देते हैं जो कि एक पॉलिसी या रिकमेंडेशन के रूप में बाहर आ सके. नमस्कार, I welcome you all to this forum of FloraFauna Science Foundation today as the Innovation Summit or FFIS 2024, which is a culmination of the web series that we run with experts from various fields related to our theme, and also your use in villages as well as urban areas through science to enable health, environment, nutrition, and agriculture, that is HENA. As you know very well, that today we have Dr. D.B.A. Narayana as our main speaker. He is going to give us the innovation lecture on a topic that will benefit not only the farmers, but the entrepreneurs, startups, and also the industry to know the in-depth things about how herbals can really steer not only secondary agriculture, but also industrial role in health sector. And that's what fulfills our HENA. I welcome Dr. D.B.A. Narayana, and I also welcome Dr. Pathak, Professor Pathak, who is Vice Chancellor of Chhatrapati Shahuji Maharaj University, and also Professor Avasthi, who is Pro Vice Chancellor in CSJMU. Our conveners, Soni and Manika, are also here. I welcome them also, and Dr. A.K. Singh, Chairman of this organizing committee, and also Vice President of FloraFauna Science Foundation. So to begin with, I'd like to take you back to FloraFauna Science Foundation itself. It's a mission, it's a thought which has been translated. ये एक ऐसी परिकल्पना है जिसको एक स्वरूप मिलता चला जा रहा है कि ये गांव-गांव जाकर गांव की पगडंडियों से सशक्तिकरण कर सके उन यंग जनरेशन का, उस नई पीढ़ी का और किसानों का और उद्यमियों का, कि वो टेक्नोलॉजी द्वारा लाभान्वित हो सके. So we try to translate what has happened in technology for the common masses, both the society, as well as the farmers. And that is the reason that FloraFauna Science Foundation has membership right from researchers and students or research scholars, scientists to farmers, entrepreneurs, startups, small industry, and also the people representing the society as such, like journalists, media people, even those who facilitate business and those who are into education sector, into teaching, colleges and schools. So such a diverse membership tells that the beneficiaries can be on all directions. Uh, Today's summit has a theme. As you all know, we always run on a theme for the whole year. And this year's theme has been Fasal se Safal. That means harvest to success. I'll not say fasal as crop, I will say harvest because crop makes meaning only when it is harvested. And the meaning of fasal goes in both the directions. But it has been a journey. Today is fifth summit, and this summit has reached Fasal se Safal through various themes, right from 2019 to 2020, and then till today. So 1920, we had started with mission FloraFauna. That is, from flora to fauna, what are the things that can be done? And we can make HENA, that is health, environment, nutrition, and agriculture work. And that is our tagline, enabling society to achieve health through science. And that science basically revolves around agriculture only. So right from primary agriculture to secondary agriculture and secondary to tertiary, and that's how the value addition keeps happening. 2021, we had a theme, Khet se Sehat. Now, Khet se Sehat was that how we can cultivate health in farms. And that is where we had innovation lecture by Professor Trilochan Mahapatra, Director General of Indian Council of Agriculture Research. And 21-22, we moved from Khet se Sehat into Poshan se Roshan. How we can really empower society through nutrition. Again, the nutrition will come from agricultural fields. And we had the innovation lecture by Dr. Mande, Director General of CSIR. And from there, then we moved into the next theme in 22-23, Krishi se Khushi. Now, this Krishi se Khushi was how do we bring happiness, prosperity through agriculture by enhancing incomes and also enabling farmers with the modern day technologies. And that is where we had the speaker, Professor V. Prakash, who is a distinguished scientist in CSIR and was director of CFTRI Mysore. And today, 23-24, we are now on the theme Fasal se Safal, where we have none other than Dr. DBA Narayana, who has worked so much into herbal sector, into the Ayurveda sector, Ayush sector, and also industry with the background in Dabur and Unilever. And he has been a key person in steering the Indian pharmacopoeia. He used to interact a lot with me when I was at CMAP to put the elements of Indian pharmacopoeia into real validation phase. So today he is going to cover those aspects which lead to the theme that we are today talking about, Fasal se Safal. Now, when we say Fasal se Safal or harvest to success, it has two components. First is harvest and second is success. Now, harvest means we have to make the harvest itself good, and therefore good crops or good ventures have to be taken up. And that can happen only we have intelligence, the market intelligence and industrial intelligence. Then the input costs have to be reduced. How do we do that? That the harvest becomes really profitable. High yields, the varieties or the plants which will give you higher amount of what is going to give you the value, and specialized crops, like we always say nutraceutical crops or medicinal crops or aromatic crops or industrial crops and so on. On the other hand, as I said, success depends on not only getting a good harvest, but also converting harvest into a valuable product. And that is where post-harvest technologies, post-harvest processing make a lot of difference. Then how do we add value to the harvested part, and also make sure that the non-usable, otherwise non-usable part of the plant or the crop is also utilized in different ways in product development and ultimately market. So brand building, even for those, can really help all villagers to become entrepreneurs and experts. And that is where some of the incubators try to work. But what we need is accelerators, and that's where FloraFauna plays a great role. So in this journey of 5 years, we have not done it alone. We had collaborations and collaborators. The collaborators with whom we had MOUs, and those who joined hands with us on different events. And very interestingly, it happens with the CSIR, where we had IITR, where Professor Alok Dhawan had initiated this collaboration with us. Now he's director of Center for Biomedical Research in Lucknow only. And IITR worked with us to make healthy society in villages through clean water. And today we are going to have another component of CSIR getting added into our MOU and collaboration, that is NBRI, National Botanical Research Institute. From ICR, we had 3 institutes who have really contributed to us in a big way in events as well as knowledge sharing. These are National Institute of Plant Biotechnology in Delhi, NIPB, Indian Agriculture Research Institute, IARI, Dr. A.K. Singh, Dr. Ashok Kumar Singh, who is director of IARI, has been very, very keen and helpful in enabling FloraFauna translate its work. And lately we had DMAP-R, that is Directorate of Medicinal Aromatic Plant Research in Anand, who have joined hands with us and are helping us. In addition to that, we had state institutes like Center for Aromatic Plants and RFRAC, that is for in Lucknow, so UP and Uttarakhand institutes. We had industry called Bharat Rohan. We have Nishant Aroma, Dr. Mr. Ramakant Harlalka, its CMD, is also member of FloraFauna Science Foundation. We have Swaraj Herbal Plants in an industry where Mr. Bhupender Singh and Mr. Saurabh Singh are members of it. We have universities. The university which is collaborating today with us, CSJMU, that is Chhatrapati Shahuji Maharaj University, Kanpur, earlier called Kanpur University, and also KMCLA, Khwaja Chishti Language University in Lucknow. And that way we have been moving on and on. And as we always say, our mission is to create entrepreneurial villages or enabled villages, e-villages. We started with Rajagada in Itonja, then we reached Ahmedpur in Raebareli, Bakshi Ka Talab, and then Barabanki and Ba in Agra. And now we are also moving into South with Dr. Sundaresan helping us in making some e-village and some schools to adopt this knowledge in Karnataka. And the approach has been, our messenger is our regular magazine, which comes every 6 months, called Flora Sarthi. Dr. A.K. Singh is its chief editor, and Mr. Sundaresan, Dr. Sundaresan is its executive editor. And we have an approach of making Nutri-Vatika in villages as well as in urban areas, where people learn about what are the plants which can give us nutrition and health, how to grow them even if you do not have land, and then Nutri Pathshala in schools, where we tell them about the portion, we tell them about nutrition, the role of nutrition, and the havoc of malnutrition which we have to avoid. And as a consequence, we want to create Nutri villages, which are happening in form of the vegetable villages, and Nutri Kisan, the Kisan who is going to take up, like we had chia and we have quinoa in Telangana and Karnataka. So that is how we are moving on. I am very grateful to everyone who has contributed to making this mission move forward. And today reaching the theme where we talk about how we are going to succeed from the crop or harvest of the crop and make it an entrepreneurial venture and make those venturists as the enabled village people. And without taking further time, I'd like to thank once again Professor Pathak and Dr. Narayana for having joined us. Also, I would like to thank Professor Avasti, who always is keen in helping us from Kanpur CSJMU for enabling our efforts in this direction. Also, I welcome all the participants who are going to be in the various sessions of today's summit, starting with this, what we call as Aarambh, moving into Khoj, that is where people will be presenting their work, and Manthan, where we will have panel experts discussing the things, and ultimately Amrit coming out of that, which will be the concluding session. Thank you, and I welcome you once again. Namaskar.
Thank you, sir. Thank you. Thank you so much. So we await to hear from Dr. D.B. Anantanarayana, our distinguished speaker of the forum. I request Dr. Soni Gupta to please introduce him.
Thank you, Dr. Manika. It's been a great pleasure to introduce Dr. D.B. Anantanarayana to all of you. And before I start the elaborate biodata, in enumerating all his achievements, let me tell you that CSJMU is also celebrating its 59th Foundation Day, and it's been a great pleasure hosting this event with FloraFauna Science Foundation. So warm greetings to all of you from CSJMU Kanpur. Dr. D.B. Anantanarayana is a pharmaceutical scientist with over 40 years of experience in research and development. He is recipient of Eminent Pharmacist Award of Indian Pharmaceutical Association in 2007. He has worked in both pharmaceutical industry and leading R&D on botanicals and Ayurvedic products, taught pharmacy, and has worked with Ranbaxy, Dabur, and Hindustan Unilever Research. He has contributed to monograph development for botanicals in IP. He championed regulations for notification of pharmaceuticals regulation, nutraceutical regulations, and Ayurveda Ahar regulations in India. Demonstration of reverse pharmacology approach with commercially successful products has been his forte, apart from leading research for several anti-cancer drugs, first time in India, and a large number of Ayurvedic herbal products as well as food products development, which have been commercially successful. He currently chairs committees in Indian Pharmacopoeia Commission and in Food Safety and Standards Authority. He has been awarded IRF IPA Lifetime Achievement Award in 2021. With this biodata, let me welcome Dr. D.B. Narayana, and I will welcome him to give his innovation lecture entitled Random Thoughts on Developing Scenarios in Cultivation of Medicinal Plants, Some Observations and Suggestions. Over to you, Dr. D.B. Narayana. Thank you, sir.
मातृभ्यो नमः, पितृभ्यो नमः, गुरुभ्यो नमः, आचार्येभ्यो नमः, सर्वदेवताभ्यो नमः. Good morning to all the devatas that are going to listen me for the next 30 minutes. Please let me know whether I should speak in Hindi or English or English.
English is the best.
English?
English.
English. Okay.
Yeah, you can mix Hindi and English. Yeah.
Yeah, very good. आज जब प्रोफेसर सुमन खनूजा और बाकी लोगों ने बताया एफएफएस फाउंडेशन जो है क्या काम कर रहा है, कितने साल से काम कर रहा है, मुझे भी बहुत सारा जानने की मौका मिला. मुझे बहुत सी चीज़ें पता नहीं था, एंड आई एम वेरी हैप्पी कि आज मुझे यहाँ पे कुछ बोलने के लिए इनवाइट की गई. और क्योंकि बहुत सारे पार्टिसिपेंट्स यंगस्टर्स एंड पोटेंशियल स्टार्टअप्स और फार्मर्स होंगे अपार्ट फ्रॉम रिसर्चर्स, मैंने जो थॉट प्रोसेस माइंड में बनाया था उसको थोड़ा मॉडिफाई करके मैं आज बात करूँगा. और ओपनली क्लियरली मैं कहूँगा कि मैं कोई कृषि वैज्ञानिक नहीं हूँ. आई एम नॉट ए फार्मर. I have not cultivated any plant on my own, but worked in companies where I promoted all this to happen. So please take whatever I say with a pinch of salt. But हमारे जो मोस्ट रेस्पेक्टेड साइंटिस्ट इंडिया में जो है डॉक्टर मशेलकर, उनकी एक स्टेटमेंट हमेशा मुझे खटकती रहती है, बताता रहता है कि पैराशूट्स और अम्ब्रेलास जो है तभी काम करता है जब वो खुला होगा, वो बंद होगा तो काम नहीं करेगा। इसी तरह से आज जितने लोग इस इनोवेशन समिट में भाग ले रहे हैं, मुझे सुन रहे हैं, बाकी लोगों को सुनेंगे और पूरे दिन का कार्यक्रम में भाग लेंगे, आप अपना माइंड और कान को खोल के रखिए, मुंह कम खोलिए, तो मेरे ख्याल से बहुत से चीजें अंदर जाएगी। और उसमें से मेरे को जो खुशी मिलेगी कि जितनी चीजें मैं बोलूंगा, ऑल ऑफ देम मे नॉट बी यूजफुल टू यू, उसमें से इवन एक या दो या तीन थॉट आपको अच्छा लगा, उसको लेके आप आगे जाने की इच्छा करेंगे, तो आई थिंक आज का हार्वेस्ट टू सक्सेस, मेरी टॉक भी सक्सेसफुल होगा, ये मेरा मानना है। तो इसीलिए मैंने टाइटल ही ऐसा दिया कि रैंडम थॉट्स है ये कोई स्पेशली ऑर्गेनाइज्ड थॉट्स नहीं है कि डायरेक्टली ये आप करिए, इससे इतना पैसा मिल सकता है या ये फलाना चीजें हो सकता, ये नहीं। बट मैं आपकी पैराशूट को खुलवाने की कोशिश करूंगा, अम्ब्रेला को खुलवाने की कोशिश करूंगा कुछ नए थॉट से। जब भी कल्टीवेशन ऑफ मेडिसिनल प्लांट्स की बात आती है या मेडिसिनल प्लांट्स का जितने अच्छे समिट्स आपने किए हैं, FFIS ने किए हैं लास्ट 4-5 ईयर से, एक बहुत बड़ा डिस्कशन होता था इंडिया में कि जो भी लैंड अवेलेबल है वो बढ़ता गया बढ़ता गया बढ़ता गया फैमिली में, और हर एक आदमी के पास छोटे छोटे टुकड़े रह गए बिकॉज ऑफ द डिविजन ऑफ द प्रॉपर्टी। बहुत सी कम लोगों के पास बहुत सारा लार्ज एकरेजेस, 500 एकड़, 100 एकड़, 1000 एकड़ लैंड किसी के पास होगा, ये बहुत ही कम एग्जांपल्स मिलेगा। मगर टेक्नोलॉजी जो लास्ट कपल ऑफ ईयर से थिंकिंग हुई है टू थिंक आउटसाइड द बॉक्स, उससे एक चीज हम सीख सकते हैं कि साइज ऑफ लैंड इज नॉट इम्पॉर्टेंट, साइज ऑफ योर माइंड एंड द वे यू थिंक इज मोर इम्पॉर्टेंट। तो ये एक थीम है जो मैं ट्राई करूंगा आज बोलने को। और मेरे हिसाब से कौन सी मेडिसिनल प्लांट्स को आप थिंक कर सकते, क्या स्ट्रेटेजी बना सकते, कैसे कर सकते, इसके बारे में आई विल गिव सम टॉक्स। प्लीज नेक्स्ट स्लाइड प्लीज। जो माइंड खुला रखा है, आज हमारे माइंड में एक पद्मश्री दिया हुआ है सर्वेश्वर बासुराम तारी को। इस फार्मर को गवर्नमेंट ऑफ इंडिया ने पद्मश्री से सम्मानित किया। ये आता है पनबरी विलेज ऑफ चिरांग डिस्ट्रिक्ट। इन्होंने अपनी दिमाग खोल के रखने से ये क्रॉस ब्रीडिंग करके और मल्टीपल प्रोडक्ट्स को, मल्टीपल चीज़ों को उगा के और इंटरक्रॉपिंग करके जो चीज़ें हासिल की, उसको उनकी विलेज में सब ने फॉलो किया और वो पूरा विलेज जो है इंटीग्रेटेड फार्मिंग में अग्रसर हो गए। और एक आदमी, कोई ज़रूरी नहीं है उसके पास PhD चाहिए या एग्रीकल्चर का डिग्री चाहिए, जो आप काम कर रहे हैं उसको खुला मन से सोच के करने से हमें कितना खुशी होता है कि ये सर्वेश्वर जी जो है इंटीग्रेटेड फार्मिंग की अपनी सोच बढ़ा के मॉडल बन गए। और कितना अच्छा है कि गवर्नमेंट जो है ऐसा ग्रास रूट कल्टिवेटर को, फार्मर को रिकग्नाइज की है। सो दिस इज वेरी वेरी इंपॉर्टेंट। इंटीग्रेटेड फार्मिंग की बात बहुत पहले हुआ करता था, इवन व्हेन आई वाज इन डाबर सोचते थे हम बैकवर्ड इंटीग्रेशन पे बहुत काम किया, आज भी डाबर काम कर रहा है कि हमारे को जो जड़ी बूटी चाहिए, मेडिसिनल प्लांट्स चाहिए, उसके पार्ट चाहिए, उसको हम खुद ही कल्टीवेट कराए। तब बहुत कम बात होती थी इंटीग्रेटेड फार्मिंग की। आजकल मोर न्यू टेक्नोलॉजीज आई है तो इसकी कम बात होती है, मगर आज भी इसका वैल्यू है, ये इसने सर्वेश्वर ने प्रूव कर दिया। नेक्स्ट स्लाइड प्लीज। तो दूसरी चीज क्या हो रहा मार्केट में, अभी तक जो है लड़ाई होती थी एलोपैथी और आयुर्वेदा या मेडिसिनल प्लांट्स बेस्ड दवाइयों पे या ड्रग्स पे। अब ये लेटेस्ट न्यूज़ है, जस्ट जनवरी 31 2024 को आप देखिए टाटा मेमोरियल कैंसर सेंटर जो है, उन्होंने 500 एकड़ लैंड उनके पास जो थी उसमें उन्होंने डिसाइड किया शतावरी, ब्राह्मी, कथा और पुष्कर मूल और अश्वगंधा जैसे प्लांट्स को ग्रो करना चाहते हैं, ग्रो करना शुरू कर दिया, because they are using these plants in their treatment for cancer patients, and they are getting lot of supporting benefit, improvement in the efficacy of the drugs that are being given to cancer patient, improvement in their mental status, improvement in their kidney functions and liver functions, improvement in their immunity levels, because most of the anti-cancer drugs जिसको दिया जाता है उसकी इम्यूनिटी कम हो जाती है, इसीलिए 3-3 हफ्ते के बाद दूसरा इंजेक्शन लगता है, लोगों की बाल उड़ जाता है और पतले हो जाते हैं, लॉट्स ऑफ साइड इफेक्ट्स कम अप फ्रॉम दोस, बट कैंसर इज सच ए डिसीज। बट जो लोग लड़ते थे कि आयुष में कुछ नहीं है, मेडिसिनल प्लांट्स जो काम नहीं करता, वो लोग आज कल्टीवेशन की बात कर रहे हैं। किस टाइप के प्लांट्स? शतावरी है, ब्राह्मी है जो माइंड पे काम करता है, कथा है जो इम्यूनिटी को बढ़ाता है और बायो अवेलेबिलिटी को एनहांस करती है, पुष्कर मूल है जो लिवर और किडनी में स्टोन्स ना फॉर्म हो, स्वेलिंग ना हो, इसके लिए सपोर्ट करती है। दे आर यूजिंग इट टुगेदर, सो दिस इज अनदर वे व्हाई पीपल आर लुकिंग एट मेडिसिनल प्लांट्स एंड क्या चीज हम कर सकते हैं। नेक्स्ट स्लाइड प्लीज। तीसरा जो है, जो फॉरेस्ट में काम कर रहे हैं, फॉरेस्ट कंजर्वेटर्स ऑफ फॉरेस्ट को बोटानिकल सर्वे ऑफ इंडिया अभी शुरुआत की है। मे बी FFIS शुड लुक एट दिस एरिया ऑल्सो एज एन इम्पॉर्टेंट एरिया। और उन लोगों को जो फॉरेस्ट में काम करते हैं ऑफिसर से, दे डोंट नो विच इज व्हाट मेडिसिनल प्लांट्स, उनका क्या नाम है, उनका आइडेंटिफिकेशन क्या है, कैसे उसको देखना है। ये फॉरेस्ट डिपार्टमेंट मिनिस्ट्री का उन्होंने बोटानिकल सर्वे ऑफ इंडिया के साथ मिलके उनको ट्रेनिंग प्रोग्राम्स कर रहे हैं, हाउ टू आइडेंटिफाई प्लांट्स एंड टैक्सोनॉमिक एस्पेक्ट्स बताए जा रहा, सो दैट ये ऑफिसर जब फॉरेस्ट में मूव करते हैं वो देख सकते कौन सा क्या मेडिसिनल प्लांट है, आइडेंटिफाई कर सकते हैं और किसकी इंपॉर्टेंस ज्यादा है उसको ज्यादा केयर कर सकते हैं। ये नॉलेज इनको दिया जा रहा है। तो देखिए कितनी बढ़ रहा है नॉलेज। तो इसका मतलब है whoever is involved in cultivating or looking at this area or harvest to success, क्या हम उगा के हार्वेस्ट कराए और क्या सक्सेस मिलेगा, इसकी जो है स्काई जो है बहुत खुल रहा है। नेक्स्ट स्लाइड प्लीज। और इस टाइप की न्यूज और एडवर्टाइजमेंट बहुत सी जगह छपती है, मनी मेकिंग एग्रीकल्चरल बिजनेस आइडियाज। कोई आदमी आज की तारीख में 3 चीजें मैंने नोट की है। एक तो फार्मर्स जो है दे आर बिकमिंग मोर कंड्यूसिव और मोर नॉलेजेबल अबाउट द कॉस्ट ऑफ कल्टीवेशन, कॉस्ट ऑफ हार्वेस्टिंग, प्रॉपर स्टोरेज, नहीं तो बिकेगा नहीं, और मार्केट में इस जड़ी बूटी का, इस चीज का क्या रेट है, मेरे को किस रेट पे बेचना है, ये नॉलेज अवेलेबल है। बिकॉज टेक्नोलॉजी और इंफॉर्मेशन टेक्नोलॉजी इतनी बढ़ गई है, पीपल आर वेरी वेरी एस्ट्यूट, और नॉलेज जो है 2 मिनट के अंदर सबके यहाँ से पता लग जाता है। तो बहुत सारे एग्रीकल्चरिस्ट और फार्मर्स और कल्टीवेटर्स जो है अकेले फार्मर नहीं है, वो दिमाग जो है इतना खुल के रखा है कि इसको कैसे मैं हार्वेस्ट करूं, कैसे रखूं और कैसे पैक करूं, और किसको बेचूं, किस दाम से बेचूं, ये सब उनको जानकारी मिल रहा है, सीखना चाहते हैं, सीख रहे हैं। और पीपल आर सो ओपन, फार्मर्स जो है इतनी एक्सपेरिमेंटल है कि अपनी पास जितनी जमीन है उसमें मे बी 20% 25% जमीन पे एक्सपेरिमेंट करते, बाकी में जो रोज उगाते हैं उगाते रहो, उससे बाद पैसा बन रहा, आपका बेसिक नीड्स पूरा हो रहा, बट मैं नया नया चीज करूंगा। लोग जो है बहुत से जगह में नॉर्थ इंडिया में स्ट्रॉबेरी कल्टीवेट कर रहे हैं, कुछ लोग जट्रोपा डाल के कल्टीवेट करते हैं। सोयाबीन का तो एंड ही नहीं है, बहुत सी जगह में सोयाबीन ग्रो हो रहा है, उसका हेल्थ एस्पेक्ट्स, न्यूट्रास्यूटिकल और फूड एस्पेक्ट्स बहुत ही बढ़ गया है। और सोया मिल्क भी बन रहा है, तो नई नई प्रोडक्ट्स आ रही है। और स्पाइसेस, स्पाइसेस में इंडिया इज बिकमिंग नेक्स्ट काइंड ऑफ ए रेवोल्यूशन इज हैपनिंग इन कल्टीवेशन ऑफ स्पाइसेस इन इंडिया। और ये बहुत इंपॉर्टेंट है क्योंकि ग्लोबली आप वेस्टर्न कंट्री में ट्रैवल करते हैं, पेपर, रोजमरी और एक या दो स्पाइसेस छोड़ के उनको कोई स्पाइस की जानकारी नहीं था। और लास्ट 15 ईयर से इंडियन करी, इंडियन मसाला ग्लोबली इतना फेमस हो गया है, दो चीज की वजह से। एक तो उनकी टेस्ट की वजह से, और दूसरा उसका हेल्थ बेनिफिट की वजह से। एवरीबडी ग्लोबली एक्सेप्ट्स कि स्पाइसेस आर सो इम्पोर्टेंट फॉर कीपिंग हेल्थ, एंड सो देयरफॉर देयर इज ए बिग बिजनेस built around cultivation of spices. Next slide please. ये एक एडवर्टाइजमेंट आजकल हर जगह छपता रहता है, नया नया कंपनीज। अब देखिए 15 एग्री टेक स्टार्टअप्स, दे आर डिसरप्टिंग एग्रीकल्चरल लैंडस्केप इन इंडिया। ये कोई ग्लोबल एक्सपीरियंस नहीं बता रहा हूँ, दीज आर ऑल स्मॉल कंपनीज, दीज आर ऑल स्टार्टअप। और इनमें मैंने थोड़ी सी इन्वेस्टिगेशन की, 90% ऑफ देम आर नॉट फार्मर्स, their parents were not farmers, many of them are IIT engineers, yeah, MBA people who have gone abroad, and somehow they didn't like what they were doing, they have come back, India वापस आ गए और सोचा यार कुछ नया चीज करना मुझे। और ये लोग स्टार्टअप शुरू किए टू कल्टीवेट इंटरेस्टिंग प्लांट्स एंड इंटरेस्टिंग थिंग, एंड बिकॉज उनकी दिमाग में मैं जो भी काम करूंगा प्रोफेशनली करूं, साइंटिफिकली करूं और सोच समझ के करूं और अच्छा करूं, और उसकी वजह से उनकी स्टार्टअप्स जो है बहुत ही सक्सेसफुल हो रहा है। फ्यू एग्जांपल्स मैं आपको दूंगा। नेक्स्ट स्लाइड प्लीज। ये एक देखिए वडोदरा का एक कपल का स्टोरी रिसेंटली छपा है। आप लोगों को एक नया टेक्नोलॉजी लास्ट कपल ऑफ ईयर से बात हो रहा है, आई एम श्योर FFIS में इसके ऊपर चर्चा हुआ होगा, कॉल्ड हाइड्रोपोनिक्स और एरोपोनिक्स। ये हाइड्रोपोनिक्स जो है, यू डोंट नीड लॉट ऑफ सॉइल, लॉट नंबर ऑफ लैंड्स। ये एक 10 बाई 10 कमरे में या 20 बाई 20 कमरे में या 60 बाई 40 साइट आपके पास है, उसमें आपने इसकी इक्विपमेंट्स लगा दी, और इंस्टेड ऑफ लुकिंग एट लार्ज एकरेजेस यू लुक एट वर्टिकल। तो उसमें स्टैंड्स बना दीजिए एट स्पेसिफिक डिस्टेंसेस, यू हैव ट्रेस, एंड ऊपर से पानी धीरे धीरे टपकती रहती है, नीचे पानी है, और जो भी प्लांट आप ग्रो कर रहे हैं वो पानी में ही ग्रो कर रहे हैं, और पानी के अंदर ऐसी मीडियास, ऐसे सॉल्ट्स, ऐसे ऑर्गेनिक सब्सटेंसेस आपको डालते हैं। और ये जो कपल का क्यों फेमस हुए, इन्होंने 10 बाई 10 कमरे में हाइड्रोपोनिक्स और एरोपोनिक्स यूज करके सैफ्रॉन ग्रो करते। अब आप जानते हैं सैफ्रॉन जो है, केसर जो है, पहले बिलीफ होती थी कि सिर्फ कश्मीर में ग्रो हो सकता और कहीं ग्रो नहीं हो सकता, और वो भी एक स्पेशल बल्ब चाहिए, बहुत केयरफुली करना है। अब इस कपल ने प्रूव कर दिया कि मैं एक छोटा सा 10 बाई 10 रूम में जो भी टेम्परेचर कंट्रोल से वो लगा दूंगा, एयर कंडीशन कर दूंगा, हॉट एयर जब चाहिए कर दूंगा, लाइटिंग कर दूंगा, और वर्टिकल ग्रो करूंगा बल्ब्स को इन स्मॉल पॉट्स, इन स्मॉल स्पेशली मैन्योर डाल के। और ये पूरी मेरी प्रेजेंटेशन आपके पास है, जिसको चाहिए प्लीज सर्कुलेट होगा, ये न्यूज़ आप पढ़िए। और ये कपल सिर्फ स्वार्थ नहीं है, और वो कह रहे कि हमें 10x10 कमरे में 3.5 महीने में जो क्रॉप मिला उसमें 1.5 लाख से 5 लाख का वर्थ सैफरॉन इन्होंने ग्रो किया। अब आप सोच लीजिए कि माइंड ओपन करके नए टेक्नोलॉजीज को लेके कैसे इन्होंने किया है। और उन्होंने ओपनली बोला है कोई और आदमी, कोई और फार्मर, कोई और साइंटिस्ट इसको मल्टीप्लाई करना चाहते हैं तो दे आर रेडी टू शेयर एंड हेल्प देम टू सेट अप सच फैसिलिटी। सी देयरफॉर a big change I would suggest in all innovators is don't worry if you don't have too large lands. You are not a farmer, you are not coming from a farmer community, you do not know anything about cultivation. If you are interested, you can learn and go vertical, not horizontal. Next slide, please. ये मंत्र है और ये YouTube में आप जाएंगे, इसको फीड करेंगे तो इन्होंने पूरी 8.5 मिनट का वीडियो बना के डाला है। इन्होंने कैसे जैसे बुक्स रखने की शेल्फ्स बनाते हैं या स्टोरेज रैक्स होता है, ऐसा 10x10 कमरे में करके वर्टिकली हाउ दे हैव कल्टिवेटेड एंड पूरी टेक्नोलॉजी उन्होंने एक्सप्लेन किया है उधर। एंड दैट इज द थिंग दैट आई वांट ऑल द पार्टिसिपेंट्स टुडे जो लिसन कर रहे हैं मुझे, आप माइंड खुला रखिए। बहुत सारे खेत खरीदने की जरूरत नहीं है, आपके पास जो छोटी जमीन है वर्टिकल सोचिए, आकाश की तरफ जाने का सोचिए, और जितना पैसा है उसमें आप इनोवेट कर सकते हैं। मतलब हमारा कन्नड़ में एक कहावत है कि जितना बिस्तर है उतना लंबा आप पैर लांग करिए, जितना चादर है उतना बिछाइए। ऐसे ही आपके पास ज्यादा पैसा नहीं है, आप इस टाइप का मॉडर्न स्ट्रक्चर जो मेटल से बना हुआ कॉस्टली होगा, कोई जरूरी नहीं है। आप घर में नल का पाइप्स आते हैं, डिफरेंट साइजेस का पाइप्स को खरीद लीजिए और उसको खड़ा करके जमीन पे, और उसमें से आप ये ट्रेस जैसा बना लीजिए और खुद बना सकते हैं। अच्छा भैया मेरे पास पाइप्स खरीदने के लिए भी पैसा नहीं है, कोई जरूरी नहीं है, आप बैम्बू से बना लीजिए। जैसा लोग मशरूम कल्टीवेशन के लिए करते हैं, ऐसे ही आप अलग अलग मटेरियल से आप वर्टिकल का जो भी स्ट्रक्चर चाहिए बना सकते हैं। और पानी को ऊपर से डालने जैसा टंकी वगैरह फिक्स कर दिया, उसकी रेट्स कंट्रोल कर दी, धीरे धीरे से पानी होगी, या उसको स्प्रे टाइप का बना दीजिए, ड्रिप इरिगेटर लगा दीजिए। मतलब यू कैन डू वंडर्स एंड यू कैन थिंक ऑफ growing so many types of plants. Next slide, please. So vertical farming, I think, should be considered in thought process. Vertical farming में बहुत से चीजें हो सकता है। सिंपल हाइड्रोपोनिक्स को मिक्स करके फार्मिंग कर सकते हैं, अदरवाइज वहां पे मेन्योर और सॉइल मिक्स करके ट्रेस बना के उसमें कर सकते हैं। वेजिटेबल्स ग्रो कर सकते हैं, फ्रूट्स ग्रो कर सकते हैं, और स्पेशल मेडिसिनल प्लांट्स भी ग्रो कर सकते हैं जो श्रब्स है, जो बहुत सारा हाइट पे नहीं जाता, जो ट्रीज नहीं है, छोटे छोटे जो है जैसा भूमियामल की है वो स्प्रेड होता है। इस टाइप के प्लांट्स आप आइडेंटिफाई कर सकते हैं जो वर्टिकल फार्मिंग में ग्रो कर सकते हैं। आप पूरा ग्रो नहीं करना चाहते हैं तो आप प्लांट्स बनाइए, इधर प्लांटलेट्स बनाइए मिलियंस में, और फार्मर्स को बेच सकते हैं आप बेस्ट क्वालिटी सर्टिफाई करके, कंट्रोल कंडीशन में बना के। तो वर्टिकल फार्मिंग से बहुत सी चीजें आप अचीव कर सकते हैं। नेक्स्ट स्लाइड प्लीज। ये नेक्स्ट आई वांट टू गिव यू सम एग्जांपल्स ऑफ व्हाट काइंड ऑफ प्लांट यू शुड लुक एट एंड कैसे उसको बेस्ट हार्वेस्ट की तरफ से ले जा सकते हैं, क्या आप कल्टीवेट कर सकते हैं। आंवला, जब मैं डाबर ज्वाइन किया था तब सब लोग बोलते थे कि च्यवनप्राश में आंवला होता नहीं है, कद्दू डाल देते हैं। बिल्कुल सरासर झूठ था ये। उन दिनों में आंवला इतना ज्यादा नहीं मिलता था, बट मिलता था। बट अभी आंवला की कल्टीवेशन इतनी बड़ी हो गई है, और आप आंवला को एज ए पार्ट रख सकते हैं। बहुत जितनी भी जमीन आपके पास है पूरे आंवला लगा सकते हैं, नहीं चाहिए कोई बात नहीं, आप इंटीग्रेटेड फार्मिंग करिए, सेंटर ऑफ द प्लांट्स जमीन पे, और जो चाहे उसका खेती बाड़ी करिए, और बॉर्डर पे पूरी आंवला की ट्रीज लगा दीजिए, और तीसरे साल में आपको आंवला की फ्रूट्स मिलने लग जाती है। यूपी में एक फाउंडेशन है जो आंवला फाउंडेशन है, आंवला की पूरी खोज करते, और आप उनसे सलाह ले सकते हैं। आप सोच के कौन सा कल्टीवर।
So, sorry for interrupting, sir. So, we cannot hear you because your voice has gone on mute, I suppose.
Know how it happened. Okay, can you hear me now?
Yes, sir, it's clear.
Sorry, I don't know how it got muted. तो ये आंवला का जैसा मेरा नॉलेज है, मैं स्टडी करता रहता हूँ, बिकॉज आई डू लॉट ऑफ रिसर्च ऑन आंवला। 40 टाइप की आंवला का कल्टीवर्स अवेलेबल है आज। किसी में बहुत हाई फाइबर्स होता है, किसी में कम फाइबर्स होता है, किसी में गुठली बहुत बड़ा हो जाता है, फ्रूट कंटेंट कम होता है, किसी में फ्रूट बहुत ज्यादा मिलता है, पल्प ज्यादा मिलता है, फाइबर्स कम है। जो आंवला च्यवनप्राश बनाने वाले चाहते हैं वो लोग तो फाइबर्स कम, गुठली छोटा, और मैक्सिमम पल्प वाला चाहेंगे। कोई पिकल्स बनाना है, मुरब्बा बनाना है, त्रिफला के लिए ड्राइड आंवला चाहिए, डिफरेंट है। तो केयरफुली आप सेलेक्ट करके बॉर्डर ऑफ आपका जमीन पे लगा सकते हैं। और इम्पॉर्टेंट है कि आप सॉइल को टेस्ट करा लीजिए। आंवला की बेस्ट फ्रूटिंग के लिए सॉइल में बोरान की मात्रा बहुत इंपॉर्टेंट है। मेक श्योर दैट यू हैव एडिक्वेट बोरान। यदि बोरान की कंटेंट बहुत कम है तो आपकी फ्रूटिंग तो हो जाएगी, बट वो गिरने लग जाएगा, कोई कल्टीवेशन और यील्ड बहुत कम हो जाएगा। उसके लिए सिंपल चीजें कर सकते हैं, जैसे ही फ्लावर्स आने लगते हैं आप पर ट्री 3 से 5 ग्राम बोरान लेके थोड़ी सी मैन्योर के साथ मिला दीजिए, और उसको ट्री का बॉटम में डाल के हल्का सा पानी दीजिए, बहुत ज्यादा इरिगेशन ना करें। और दूसरी इंपॉर्टेंट है कैल्शियम। तो इसी तरह से 1 to 4 ग्राम कैल्शियम पर ट्री you give, it will help to give best quality amla fruit which has a longer shelf life also, जल्दी वो खराब नहीं होते। तो आंवला जो है मुरब्बा के लिए बिकता है, पिकल्स के लिए बिकता है, त्रिफला का ड्राइड आंवला के लिए बिकता है, और आंवला पिष्टी बनाने के लिए बिकता है। और आंवला का इतना ज्यादा मेडिसिनल प्रॉपर्टीज है, इट इज लाइक ए कल्प वृक्ष, and all kinds of benefits are available, so you can consider multiple uses. Next slide please. हमने बहुत साल पहले एक पेपर पब्लिश किया था इन जर्नल ऑफ बायोडाइवर्सिटी, बायो प्रोस्पेक्टिंग एंड डेवलपमेंट। ये पेपर आपको इंटरेस्टेड है तो मैं बाद में सर्कुलेट करा सकता हूँ। इसके लिए हमने एक ग्रिड डेवलप किया था जो मैं आपसे शेयर करना चाहता हूँ। नेक्स्ट स्लाइड प्लीज। ये ग्रिड है, आप देखेंगे वाई एक्सिस में डिफिकल्टी ऑफ कलेक्शन है, और एक्स एक्सिस में डिग्री ऑफ सस्टेनेबिलिटी एंड नॉन रिन्यूएबल नेचर ऑफ बायोरिसोर्स। जब आप कौन सा प्लांट कल्टीवेट करो, किसको अच्छा हार्वेस्ट हो सकता, कौन सा बेटर रिन्यूएबल है, विच विल गिव मी मोर मनी। और जैसा आजकल बहुत सारे एडवर्टाइजमेंट होता है, म्यूचुअल फंड में इन्वेस्ट करो, सही है, लॉन्ग टर्म आप पैसा रखना चाहते हो, तो यू कैन सेलेक्ट ए प्लांट डिपेंडिंग ऑन हाउ क्विकली यू वांट टू हार्वेस्ट and get success, or how patiently you are ready to wait for long time. इसमें क्वाड्रेंट 1 है, 2 है, 3 है, 4 है। क्वाड्रेंट 1 जो है वो मोस्ट इजीली एक्सेसिबल एंड रिन्यूएबल एंड सस्टेनेबल। ये लीव्स एंड स्टेम एंड स्मॉल फ्रूट्स एंड फ्लावर्स वो क्वाड्रेंट 1 में आता है। और क्वाड्रेंट 2 में रीजनेबली डिफिकल्ट टू एक्सेस एंड मीडियम स्केल, तो थोड़ा सा डिफिकल्टी होगी, ज़्यादा देर वेट करना होगा, कल्टीवेट करने के बाद उसको हार्वेस्ट करने के लिए भी आपको बहुत सारी चीज़ें सोचना पड़ेगा, बिकॉज यू नो व्हेन यू आर ट्राइंग टू हार्वेस्ट ए फ्रूट और ए फ्लावर और ए लीफ, मोस्ट ऑफ द टाइम वो नीचे गिर जाता है ज़मीन पे गिर गया तो वो कंटेमिनेट हो जाती है, उसको सफाई करना मुश्किल है, ड्राई करना मुश्किल है, उसके लिए भी एक नई नई टेक्नोलॉजी की ज़रूरत है। क्वाड्रेंट 3 जो है डिफिकल्ट टू कलेक्ट एंड डिफिकल्ट टू रीजनरेट, जैसा स्टेम है, बार्क है, ये डिफिकल्ट टू कलेक्ट है। क्वाड्रेंट 4 तो अति मुश्किल, कलेक्शन के लिए भी डिफिकल्टी है, वो रूट है, रूट बार्क है और ट्री का हार्ड पार्ट है। तो व्हेन यू, आई वुड रेकमेंड कि इस टाइप का ग्रिड आप माइंड में बना लीजिए, और आप जो प्लांट सेलेक्ट कर रहे हैं, जिसको हार्वेस्ट करना चाहते हैं, सक्सेस की तरफ जाना चाहते हैं, सोचिए कि कहाँ ये बैठेगा। उसके बाद आप डिसाइड करिए कोई बात नहीं, मैं क्वाड्रेंट 4 में काम कर रहा हूँ, 2 साल के बाद मुझे पैसा मिलेगा, मगर इतना ज्यादा सक्सेस होगा, आई एम रेडी टू इन्वेस्ट। कोई जल्दीबाजी में है, नया नया स्टार्टअप पे पैसा आता रहेगा तो तब मैं इन्वेस्ट करूंगा, तो आप क्वाड्रेंट 1 की प्लांट ढूंढिए ताकि क्विकली आपको सक्सेस मिल जाए, क्विकली आप हार्वेस्ट कर सकते हैं। इस पेपर में ईच क्वाड्रेंट का कुछ कुछ प्लांट्स का भी एग्जाम्पल है। आफ्टरवर्ड्स आई विल सर्कुलेट एंड यू कैन सर्च द पेपर ऑल्सो एंड गो थ्रू दैट। नेक्स्ट स्लाइड प्लीज। फ्यू एग्जाम्पल्स आई विल गिव यू। हाई वैल्यू प्लांट्स आप सोचना चाहते हैं तो लैवेंडर है, डिफिकल्ट टू कल्टीवेट बट हाई वैल्यू देगा आपको। अरेकनट, difficult to cultivate, it takes long time to grow, the tree has to grow, and but crop तो कैश क्रॉप है अरेकानट तो। बीटल लीव्स, अरेकानट ट्री यदि आपने डाला है उसके बगल में पान के पत्तों का लगा दीजिए, वो उसी के ऊपर क्लाइंब करता रहेगा और अब पान भी मिल गया, और जो वो सुपारी भी मिल गई आपको, बोथ आर कैश क्रॉप्स उसी ट्री में, बिकॉज वहां पे छाया होता है। पेपर आप डाल सकते हैं, पेपर या पीपली, दे आर ऑल्सो क्लाइंबर्स। कुछ ट्रीज में आप पान के पत्ते लगा दीजिए, कुछ में पेपर लगा दीजिए या पीपली लगा दीजिए, बोथ आर कैश क्रॉप्स एंड दे नीड सिमिलर काइंड ऑफ सॉइल, सिमिलर काइंड ऑफ वाटरिंग एंड पार्टली शेडी पार्टली सनी काइंड, आप स्टडी करके लिख सकते हैं। दूसरा प्लांट, आजकल कर्नाटका में बहुत सारी कंपनियां आपको प्लांटलेट्स देती है, टेक्नोलॉजी देती है और हार्वेस्ट करके आपको पैसा दे देता है, वो है कोलियस फोर्सकोली। डॉक्टर खनुजा कैन एक्सप्लेन दिस प्लांट वेरी वेल। इसके अंदर एक फोर्सकोलिन करके कंपाउंड निकलती है जो ग्लूकोमा में काम आता है। इट्स एन अप्रूव्ड ड्रग, इट्स ए वेरी कॉस्टली प्रोडक्ट एंड ए बिग मार्केट ग्लोबली। सो कोलियस फोर्सकोली कल्टीवेट हो रहा है। आप सोचिए, इट इज ए श्रब, 6 महीने से 8 महीने के अंदर आपको क्रॉप मिल जाएगा और उसके बाद आप दोबारा क्रॉप डाल सकते हैं। सैफरॉन का तो मैंने एग्जांपल आपको दिखाया। नटमेग, टाइम लगता है इसमें फ्रूट्स आने में, बट नटमेग इज अ वेरी कॉस्टली एंड वेरी इंपॉर्टेंट आयुर्वेदिक हर्ब एंड वेरी इंपॉर्टेंट प्लांट। अकरकरा, अकरकरा जो है मेन इंग्रेडिएंट है इन 20 लेखियम्स इन आयुर्वेदा, और ये इंडिया में कल्टीवेट ही नहीं हो रहा था, पूरा का पूरा इंपोर्ट हो रहा था, but Dabur has successfully found the technology and they are getting it cultivated. So you can think of, it's a very important immunomodulator and a very important herb. Ruem memori, रेवनचिनी बोलते हैं इसको। रेवनचिनी जो है एकमात्र हर्ब है जो लिवर में हेपेटाइटिस बी, हेपेटाइटिस सी, हेपेटाइटिस ई, हेपेटाइटिस ए, चारों को धो के बाहर फेंक देता है। जब हम डाबर में काम करते थे, इस पे काम किया था, हमने एक पेटेंटेड प्रोडक्ट बनाया था कॉल्ड न्यू लिव फिट, उसके अंदर रोए मेमोडी डाला है रेवनचिनी। Revanchini is proven scientifically to clear and kill hepatitis B, E, partly C and A. तो it has got a very big market. वायरल को पूरा निकाल देता है. सैंडलवुड आप ये क्वाड्रेंट 4 में आता है, लंबा वेट कर सकते हैं आप. ट्रीज डाल के भूल जाइए, 8-10 साल के बाद आपको मिलेगा, मगर इसमें लीगल परमिशंस लेने होते गवर्नमेंट से टू कल्टिवेट देम. स्टीविया, everybody knows stevia. People are not looking at ajwain. I think ajwain is another very important cash crops, क्योंकि छोटी सी प्लांट है और उसकी सीड्स होता है, सीड्स छोटा छोटा साइज का होता है, टनों में या किलोग्राम्स में कल्टीवेट करना मुश्किल है, मगर वैल्यू बहुत हाई है. और आज जो है अजवाइन का एंटी डायबिटिक एक्टिविटी हैज बीन प्रूवन बियॉन्ड डाउट. तो बहुत सारे डायबिटिक्स जो इंडिया में नंबर्स बढ़ रहा है, उनके लिए अजवाइन इस्तेमाल करना बहुत ही इंपॉर्टेंट है. इट विल हैव ए वेरी बिग फ्यूचर मार्केट. आपकी जमीन है, बॉर्डर पे नीम ट्री लगा दीजिए. फ्लैक्स ट्री जो है, फ्लैक्स सीड आपने सुना होगा, और उसका तो फ्लैक्स का तो लीफ को खाते हैं एज ए वेजिटेबल. उसकी फ्लावर से चटनी बनता है, पिकल्स बनता है और सॉस बनती है, और उसका जो फाइबर्स मिलता है उसमें से धागे बनते हैं. तो ये कल्प वृक्ष है फ्लैक्स सीड. हर पार्ट ऑफ द प्लांट इज बेनिफिशियल. यू कैन थिंक ऑफ दैट. आंवला के बारे में मैं बता चुका हूँ. मोरिंगा, मोरिंगा इज अ वेरी वेरी इंपॉर्टेंट प्लांट. मोरिंगा जो सहजन बोलते हैं, इसका पत्ते को खाते हैं और पॉट्स को खाते हैं. क्यों इतना इंपॉर्टेंट है? मोरिंगा का लीफ जो है, one of the few plants which has natural folic acid in that, in high levels, and it's very important. And it is slightly bitter in taste, but cleans up the stomach. Total toxins को निकाल देती है बॉडी से. और I don't know how many of you know, इंडिया में एक कंपनी है called moringa.com. ये कंपनी उनकी सारी प्रोडक्ट्स में सिर्फ मोरिंगा लीफ या मोरिंगा पार्ट्स या फ्रूट से प्रोडक्ट्स बनाए, इंक्लूडिंग फॉर स्किन केयर एंड फॉर ईटिंग कैप्सूल्स, टैबलेट्स, बहुत सारे प्रोडक्ट्स बनाए. एंड इट्स बिकमिंग ए बिग न्यूट्रास्यूटिकल एंड सप्लीमेंट. टैमरिंड, टैमरिंड तो आप हर चीज जानते हैं, बॉर्डर्स में लगाने से. I know of some people who have about 15 tamarind plants in their farm, small farm, and साल में जब वो फ्रूटिंग आता है, 1.5 लाख से 2 लाख रुपया में लोग आके वो खुद ही फ्रूट्स निकाल के ले जाते हैं. आपको कोई पैसा नहीं देना पड़ता उसका. So it is like a cash crops. Dr. Kanuja spent whole of his life on medicinal plants, and I'm sure many others have worked on them. Distilled oils जो है, बहुत ही important market develop हो रहा है, both for perfumery and for health benefits. So you can think of some essential oil bearing plants. दवना है, भृंगराज है, basil है, thyme है, mint है, patchouli है. The list can be endless. So these are only some random. You can think of more plants and select accordingly, and all of them give you good success if you just take care of the agricultural aspects and scientifically learn what is to be done, what is not to be done, and they are all good crops to look at. Next slide, please. Lastly, I want to say that this area has not just been private investments are coming with startups and everything, but government is promoting this through National Medicinal Plants Board. Medicinal Plants Board का वेबसाइट में आप जाइए तो ये सेंटर सेक्टर स्कीम्स अनाउंस्ड है उसमें, और 3 टाइप की स्कीम है जो कल्टीवेशन ऑफ मेडिसिनल प्लांट्स के लिए सब्सिडी देती है. और इसमें 3 टाइप की सब्सिडीज मिलती है. देयर आर 55 प्लांट्स जिसका लिस्ट उन्होंने डाला है, वेबसाइट में है, जिसको 30% सब्सिडी देते हैं. आपकी जो भी कॉस्ट ऑफ कल्टीवेशन होगा उसका 30% आपको सब्सिडी गवर्नमेंट देगा, मिनिस्ट्री ऑफ आयुष देगा। 27 प्लांट्स है जिसमें 50% सब्सिडी देता है, बिकॉज ये मोर डिफिकल्ट है, डिफिकल्टी इन सस्टेनेबिलिटी है, ज़्यादा टाइम वेट करना पड़ता, देर आर टेक्नोलॉजिकल इश्यूज देयर। और देर आर 13 प्लांट्स जो 70% सब्सिडी दिया जाता है। So you can even think of this and look at them if you think आपकी जगह में, जहाँ जगह है, जहाँ जमीन है, जहाँ वातावरण कैसे है, इसमें से कोई प्लांट आप ग्रो कर सकते हैं, तो आप अप्लाई कर सकते हैं नेशनल मेडिसिनल प्लांट्स बोर्ड का, उनकी स्कीम के अंदर, एंड दे विल गिव यू मनी टू कल्टीवेट एंड आपको सब्सिडी मिलेगा। सो गवर्नमेंट भी इसको बहुत प्रमोट कर रही है। 2 साल पहले का एनुअल बजट में गवर्नमेंट ऑफ इंडिया ने 6,000 करोड़ नेक्स्ट 3 ईयर में सब्सिडाइज करने के लिए अप्रूव किया है टू प्रमोट कल्टीवेशन ऑफ आयुर्वेदिक मेडिसिनल प्लांट्स, बोथ कैश क्रॉप्स एंड फॉर मेडिसिन। एंड हर्बल इंडस्ट्री एंड न्यूट्रास्यूटिकल इंडस्ट्री इज ग्रोइंग वेरी फास्ट इन इंडिया, क्योंकि कंज्यूमर चाहता है कि मैं कम दफा डॉक्टर के पास जाओ। आई डोंट वांट टू गो टू डॉक्टर एवरी टाइम। अपनी सेहत को मैं खुद ही क्यों नहीं मेंटेन करूं, इसके लिए वो डाइट के थ्रू, सिंपल मेडिसिन के थ्रू और हर्ब्स एंड न्यूट्रास्यूटिकल्स एंड सप्लीमेंट्स के थ्रू हो रहा है। और ये जो चेंज हो रहा है वेलनेस सेंट्रिक हेल्थ केयर से इलनेस सेंट्रिक हेल्थ केयर से वेलनेस सेंट्रिक हेल्थ केयर में बदलाव हो रहा है। I am sure each one of you are consumers. You will also want to avoid going to a doctor. पहले तो अपॉइंटमेंट लेना है, वो 10 तक टेस्ट कराने को लिख लेंगे, फिर तकलीफें बहुत होगी, फिर एलोपैथिक ड्रग्स है, अपनी साइड इफेक्ट्स है, तो एवरीबडी थिंक्स कि मैं इसको प्रिवेंट कर लूँ। और सप्लीमेंट्स की मार्केट बेस्ड ऑन हर्ब्स यूसेज and their extracts usage are growing, so there is a growing market for that. Next slide please. So these are the examples that I told you about the Ministry of Ayush giving subsidies. Next slide please. आप के माइंड में एक क्वेश्चन आएगा, सवाल आएगा कि भाई मुझे कैसे पता चलेगा कि कौन सा प्लांट का क्या डिमांड है मार्केट में। तो दिस डाटा इज नॉट अपडेटेड बट डिफिकल्ट टू गेट, बट ट्रांसडिसिप्लिनरी यूनिवर्सिटी जो बेंगलोर में है, वो नेशनल मेडिसिनल प्लांट्स बोर्ड के साथ मिलके एक किताब छापी है 2017 में। इसकी नेक्स्ट एडिशन आने वाला है, ये सुना था मैंने, पता नहीं कब आएगा। मेडिसिनल प्लांट्स इन इंडिया, एन असेसमेंट ऑफ देयर डिमांड एंड सप्लाई। बहुत ही बढ़िया किताब है ये, बहुत ही ऑथेंटिक किताब है, बेस्ड ऑन ए लॉट ऑफ सर्वेस एंड क्वांटिटेटिव studies and so many aspects they have done. And ये D.K. वेद साहब और गोराया साहब ने लिखा है। D.K. वेद साहब was one of the foremost conservator of forests and then he worked in transdisciplinary university in Bangalore. Of course वो आज की तारीख में हमारे साथ में नहीं है, बट बहुत ही अच्छा किताब है, इसको आप रेफर कर सकते हैं। और इसमें से इवन दो इट इज ए 2017 डाटा, तो यू वुड गेट अ फेयरली गुड आइडिया कि कितनी टन्स की क्या डिमांड है। और दूसरा चीज आप कर सकते हैं, DGFT का वेबसाइट में आप जाएंगे, कौन सा मेडिसिनल प्लांट्स आप ग्रो करना चाहते हैं उसकी यदि आपके पास HS कोड है, HSN कोड है, उसको आप यदि फीड कर सकते हैं, उस प्लांट का उस कोड से आपको डाटा मिल सकता है कि हाउ मनी टन्स ऑफ दैट मेडिसिनल प्लांट हैज बीन एक्सपोर्टेड इन द लास्ट कपल ऑफ ईयर्स। तो दैट आल्सो इज अ वेरी गुड डाटा टू सपोर्ट योर थिंकिंग क्वांटिटेटिवली एंड प्रोफेशनली। नेक्स्ट स्लाइड प्लीज। सो मेरी आखिरी स्लाइड है ये। समरी सजेशंस क्या है? माइंड खुला रखिए, कान खुला रखिए, कम बोलिए और काम पे लग जाइए। बहुत सा चीजें हो सकता है, लर्न करिए। एंड डोंट वरी इफ यू डोंट हैव लार्ज पीस ऑफ लैंड। अरे भैया हमारे पास 100 एकड़ का जमीन नहीं है, कोई जरूरी नहीं है। आज तो छोटी छोटी जगह में वर्टिकल नए नए टेक्नोलॉजीज आ रहे हैं, आप भी सोच के टेक्नोलॉजीज को इम्प्रूव कर सकते हैं। और उसमें डिपेंडिंग ऑन अवेलेबिलिटी ऑफ फाइनेंस यू कैन मेक इट। एंड छोटे छोटे यंगस्टर्स को आप पकड़ लीजिए, वो टाइमर लगा देंगे, पंप को ऑन ऑफ, लाइट को ऑन ऑफ सब सेट कर देंगे, और एनर्जी भी सेविंग होगा, वाटर भी कम यूज होगा, रीयूज होगा, बहुत सी चीजें कर सकते हैं। और कोई बाउंड्री मत रखिए माइंड में। वर्टिकल स्पेस का आप सोचिए, टू इंटीग्रेट इट विथ, इफ यू हैव सम हॉरिजॉन्टल स्पेस। नए टेक्नोलॉजी के लिए ओपन रहिए, डोंट थिंक कि पुराने के जमाने की तरफ से वही फार्मर्स की चीजें करना है मुझे, नहीं। उसमें हम क्या नया चीज कर सकते हैं सोचिए, और ऑटोमेट वेयर पॉसिबल। कोई जरूरी नहीं है सारा दिन लगा रहे। जैसा मैंने कहा, सिंपल टाइमर्स फॉर लाइट ऑन एंड ऑफ, स्प्रेइंग वाटर, ड्रिप इरिगेशन, सो मेनी थिंग्स यू कैन डू। एंड वर्क इन ग्रुप्स, अकेले में मत काम करिए, क्योंकि नॉलेज और डिफिकल्टीज को भी शेयर करिए, और हैप्पीनेस जो है बढ़ जाएगा। क्योंकि एक दूसरे से कोऑपरेटिव सोसाइटीज यदि आप बना सकते हैं, 20 फार्मर, 40 फार्मर, 10 टेक्नोलॉजिस्ट मिलके करिए, इन्वेस्टमेंट कम होगा और उपलब्धियां बढ़ जाएगी। और जो भी करना चाहते हैं, पहले एक छोटा सा एक्सपेरिमेंट करिए। आपकी खेत में 1/10 1/20 जगह आइसोलेट करके वहां ग्रो करके देखिए, 1 साल 6 महीना। और इसमें मोस्ट इंपॉर्टेंट है, हर दफा सोचते हैं फर्टिलाइजर, ऑर्गेनिक भी कर सकते हैं। और सिंपल चीज है, मैंने जो सीखा है, सुना है, बहुत सारे सॉइल में नाइट्रोजन की कमी हो जाती है। एंड एवरीबडी थिंक्स ऑफ गोइंग एंड बाइंग NPK डाल दो, नाइट्रोजन डाल दो, ये डाल दो। कोई जरूरी नहीं है। एक दफा मूंगफली ग्रो कर दीजिए, वहां पे मूंगफली लगा दीजिए, और 3 महीने में आपको मूंगफली भी मिल जाएगी खाने के लिए। और इट फिक्सेस नाइट्रोजन इन द सॉइल। ग्राउंडनट इज द बेस्ट प्लांट टू फिक्स नाइट्रोजन इन द सॉइल। एक क्रॉप आप मूंगफली ले लीजिए, इंटीग्रेटेड क्रॉपिंग कर दीजिए, लुक एट योर लैंड, इट वुड बी रियली वेरी गुड। एंड मोस्ट इंपॉर्टेंट इज, जब भी आप कोऑपरेटिव बनाएंगे, ग्रुप में करेंगे, स्टोरेज के लिए जगह ढूंढिए और प्रॉपर स्टोर करिए उसको। अदरवाइज उसी में पेस्ट हो जाएगा, फंगस ग्रोथ हो जाएगा, बैक्टीरियल ग्रोथ हो जाएगा, स्मेल चला जाएगा। तो स्टोरेज स्पेस को भी पहले ही आइडेंटिफाई करिए। मैं उगा दिया, मैं कहाँ इसको स्टोर करूंगा। आजकल बहुत से जगह गवर्नमेंट सपोर्ट से कोल्ड स्टोरेज बन रहा है, स्टोरेज के लिए बन रहा है जगह, आप उसको रेंट कर सकते हैं। आपकी पैदावार को और हार्वेस्टेड मटेरियल को प्रॉपरली स्टोर करिए, पैक करिए। एंड दिस आई वुड से धन्यवाद आपको। मैंने जो समझा उसको आपको शेयर किया। थैंक यू फॉर लिसनिंग टू मी। और वट एवर इज यूजफुल टू यू, यू प्लीज टेक। जो नहीं चाहिए उसको फेंक दीजिए। एंड आई थैंक FFIS and all the people to invite me, to give me this opportunity to share my thought. Thank you very much.
Thank you so much, sir. Thank you. Thank you for that enriching talk. We would definitely work on your suggestions, like collaborating with the forest department in context of maps, new capacity building programs on hydroponics, as you said, and small scale modern agriculture ventures like vertical farming and all. Thank you again for sharing all these insights with us. And of course, we would like to read the paper. That's why I've shared the paper among the group in the chat box. So all of you who want to read that paper sir mentioned, you can go in the chat box and download that. So thank you so much, sir. Thank you.
Oh, it's my pleasure.
Thank you, sir. And moving forward, it's a matter of great delight that we have signed an MOU, as Kanuja sir said in the beginning of the meeting, with CSIR National Botanical Research Institute. So for that, I would request Dr. Neeraj Jain, General Secretary, FFSF, and Dr. A.K. Singh, Vice President, FFSF, to please coordinate.
थैंक यू डॉक्टर मनिका। जैसा बताया कि हमारे लिए प्राउड मोमेंट है पर फ्लोरा फौना साइंस फाउंडेशन की हम
Sir, if you permit, can I leave?
Sir, yes, sir.
Sir, before you leave, we would like to, sir, please
Just
give
wait
us
for
30
a couple
seconds.
of minutes.
I mean,
Wait for
sir.
a couple of minutes.
So just give me a few seconds since I was not.
Yeah.
No issue because I have to go somewhere. So,
Okay. Sure. Sir, just one
Monica,
second.
before you do that, so what we'll
Yeah.
do is we'll go for citation, but before that, I just say a word.
Yes, sir.
Dr. Narayana, truly grateful to you. You have totally justified what your title says. And believe me, we all are now paragliding. We have opened our minds and thoughts, and it really has been such a wonderful talk where not only farmers but the next generation of students is going to get benefited, and definitely will like in future to keep taking guidance from you. And also many of the participants today with whom we will share your email ID might write to you. Thank you so much.
yeah, एक चीज मैं कहना चाहता हूँ. When I retired in 2010 from Unilever, before that I thought कि इतनी चीजें हमने सीखी है और सोसाइटी को, भारत देश को, इंडिया को आगे बढ़ाना है. So I made a small trust called आयुर्वेदिया ट्रस्ट. हमारे पास कोई करोड़ों रुपया तो नहीं है, छोटी सी अमाउंट ऑफ मनी है जो क्लाउड सोर्सिंग से है, मगर थॉट और आइडियाज जो है टनों में है. तो कोई भी साइंटिस्ट कोई रिसर्च कर रहा है या कोई काम कर रहे हैं, कोई डिस्कस करना चाहते हैं मेरे से, आई वुड बी वेरी हैप्पी टू गिव देम सम सजेशंस. एंड स्पेशली मास्टर्स डिग्री स्टूडेंट्स, पीएचडी स्टूडेंट्स, रिसर्च स्कॉलर्स कभी कभी फंस जाते हैं, कोई पर्टिकुलर टेस्ट वो नहीं कर पाते हैं, फैसिलिटी नहीं होता. यदि हमसे हो सकता है वो टेस्ट का खर्चा हम स्पॉन्सर करके उनको दे सकते हैं. सम सपोर्ट, छोटी छोटी चीजें मैं करता रहता हूँ ट्रेनिंग्स के लिए. So you can tell your people that if they are, one can talk to me and we can decide a date and time and we can talk. Another technology that I want to sort of share here very quickly, there is a paper published on that also. आजकल इंडस्ट्री जो है पाउडर्ड हर्ब्स को डायरेक्टली खरीदने की इच्छा ज़्यादा करता है, बिकॉज ग्राइंडिंग इज वेरी कॉम्प्लिकेटेड एंड जीएमपी इश्यूज आर इन्वॉल्व. तो हमने प्रोफेसर विधु एरी जामिया हमदर्द के साथ मिलके एक टेक्नोलॉजी डेवलप किया है कॉल्ड rightherb.in. इसमें माइक्रोस्कोपिक कैरेक्टर्स को एक डाटाबेस बनाया है. 300 प्लांट्स का, पाउडर्ड हर्ब्स का माइक्रोस्कोपिक डाटाबेस है. एनीबडी कैन गो टू rightherb.in and if they have an unknown powder, there is a SOP how to make a slide and identify parts, and जो data available है, which feature you are seeing, which you are not seeing, आप कीन करके सर्च कर दे, it will give you the result which plant it is.
Sure, we will do that. We will facilitate, catalyze it. And also, I take this opportunity to say that FloraFauna Science Foundation would definitely like to collaborate with your trust to link up the people, who could be benefited.
Yeah. Thank you, sir. Thank you very much.
Thank you, sir. So would like to share something. And sir, can we share your email ID with the participants and whosoever contacts us later? Yeah.
DRAN.
Yeah.
So we have your email ID. We'll share it with them.
Please
So
share.
we would like to give you a citation here, a small recognition.
Thank you so much. Thank you.
To honor you as distinguished member of FloraFauna Science Foundation.
Thank you very much.
Welcome, sir.
Thank you, sir. Thank you, sir.
Thank you, sir. Thank you.
It's
Thank
just
you,
a
sir.
small
Thank you.
token of appreciation.
Yeah,
Thank you,
very
sir.
much. Thank you.
So we move forward. Thank you, sir. So we move forward. Over to you, Singh, sir.
Thank you. So, next program, that is the exchange of memorandum of understanding signed between CSIR and DRI and FloraFauna Science Foundation. जैसा मैं आपको बता रहा था कि ये बड़े गौरव की बात है हमारे फाउंडेशन के लिए कि हम इस एग्रीमेंट में इंटर कर रहे हैं और ये 2 दिन पहले यानी कि 8 फरवरी को साइन हुआ है. From NBRI side, Controller of Administration, Mr. Pradeep Kumar, and from FloraFauna Science Foundation side, our General Secretary, Dr. Neeraj Jain, who is present on the panel, she signed the agreement. Today, in this important function, we are taking this opportunity to just formally exchange this MOU in presence of Director of NBRI, Dr. Sasni. If he is present, please come on the panel, please. Dr. Ajit Sasni, he is there. I could see Dr. Manish Bhair, Principal Senior Scientist of NBRI, he is there. Dr. Manish Bhair and Mr. Pradeep Kumar, COA of NBRI, if he is present, please join us. So today we will be exchanging it, and Dr. Manika, can you share the agreement?
Yes, sir. Yes, sir.
Yeah. In the meantime, I must tell you that this agreement was signed, and which has got, I mean, a very important operational part, which I am just reading. CSIR NBRI and FloraFauna Science Foundation scientists or the members will help each other in preparing common research programs, village-oriented entrepreneurship development program, particularly in the area of floriculture. That is very important area of NBRI, and they have all the strength and expertise in floriculture. So we will work in taking the technology into the field jointly with NBRI. And another important area in which we will be collaborating, we will be having joint knowledge platform for organizing thematic events, including seminars, awareness camps, training workshops, and orations on the topics of common interest. So here it is the MOU, which was inked between NBRI and FloraFauna Science Foundation. Let us go to the first page. Yeah, this start here and then scroll down. Okay. Second, and finally the third one. Thank you.
So,
And
thank you.
I think Dr.
So let's not go into financial modalities. We can share it up.
No,
to you.
Yeah. No problem. So
the final page, last page.
Yeah.
Last page, just so the last page.
Okay,
and,
last page.
And then we can have remarks by Dr. Sasini if he is present on the panel.
Sir, may I? This is Dr. Manish.
Okay,
But
Dr. Manish, yeah,
yes,
please.
actually, sir, Shastri sir is not here. He's outstation somewhere in important meeting actually.
No
So
problem.
we are on behalf of Dr. A.K. Shastri, I'll just present. This is Dr. Manish Bhoyer, working as a senior scientist and head technology transfer and business development division of CSR National Botanical Research Institute. Actually, we are grateful to means by signing this MOU, and we definitely work together in the field of floriculture as well as plant-based research with the FloraFauna Science Foundation, and whatever be the requirements regarding our assistance required for this collaboration, we will be there in future with you. Thank you very much, sir.
Thank you, Dr. Bhatt. Dr. Jain, few words.
Sure, sir. Thank you, Dr. Singh, and everyone, a very good morning to you all. This is a very important addition in the line of collaborations that we have been doing with other institutes for Science Foundation. And this is a very important institute that works in the field of plants and also in the field of agriculture, So as Dr. Boyar said, that we are going to work together in a collaborative manner in the field of plants and also the agriculture field. And we both would like to work in a mutual understanding situation wherein the vision and mission of both the organizations are taken into considerations. We are working towards these objectives, and we would definitely a very great association with a very reputed institute that we have. So it's a very great addition to FloraFauna Science Foundation. And we are very thankful to Dr. Ekashashni and also his team for steering this collaboration. Thank you.
Our founder, chairman, Sir Khanuja would like to speak something.
Thank you. Well, it's another cornerstone, the tag that has been added to the working of FloraFauna Science Foundation. Thank you, Manish, for being here to represent Ajit, and also Neeraj for representing FloraFauna Science Foundation for this agreement. We are looking forward to a very dedicated and focused collaboration wherein starting with Lucknow and around all the districts, we will like to spread the fragrance of floriculture. As you know, Manish, we have links in various villages around here, and we will like to take your teams there to equip the farmers as entrepreneurs.
Yes.
I have seen many technologies of NBRI for so many years, right from Dr. Pushpangadan's time, and particularly dried flowers. Is such a thing that a farmer would be happy to really do on it, but also your hydroponic and aeroponic system for floriculture, which can add to the cut flower industry. And that is the direction in which we are thinking to work. And also, whatever NBRI would like to guide us, that this is the new opportunity, we would like to do that. Thank you. I wish this collaboration to be a great success and set as a model.
Yes, sir.
Thank you, everybody, and Dr. Manish for sparing your valuable time for this event. Namaskar, Dr. Manika Park, next program.
Yes, sir. Thank you, sir. So at this juncture, we await the remarks by Professor S.K. Awasthi, Pro Vice Chancellor, CSJMU, on the summit scope and collaboration with FFSF. Professor S.K. Awasthi, sir.
Please unmute, sir. Unmute.
Sir.
प्रोफेसर अवस्थी, आपको अनम्यूट करना।
प्रोफेसर अवस्थी, आपकी आवाज नहीं आ रही, इसको आप अनम्यूट कर लें। हाँ, ठीक जी।
A very good morning to all of you who are participating in this conference FFIS2. I really want to welcome our FloraFauna Science Foundation Chief, Dr. Khanuja, who has been associated with us, with our university in a great manner. We are in process of formulating some plannings to actually go ahead with the objectives of the FloraFauna Science Foundation and the university together. We are a big university. Some 10,000 students are studying in the campus, including the students of pharmacy, life science, biotechnology, and agriculture sciences, which has been recently opened. So as far as the objectives are concerned, we will look into it, and we are seeing that how we can manage the objectives of this FloraFauna Science Foundation. First thing which I want to say in this conference, Fasal se Safal. Actually, our farmers are facing a lot of problems in the sense that they actually do a lot of labor in having their crops And because of the climatic conditions, sometimes it is not possible. And sometimes when it becomes possible also, they do not get the price which should be given to the farmers. So ultimately the richness or the samriddhi, what you can say, or what you can say, the Safal farmer is not being, they are not coming into the picture and more arena. So what I feel that there should be ample emphasis should be given to the conditions in which farmers are actually working. And our student strength is too much, and we want to collaborate directly with the farmers. In this sense, we can open up with some startups, and those products which are not supposed to be very useful nowadays, but can be made useful if the involvement of the students and the faculty of our university is involved. So those useful products may be obtained from the crops, and the benefit of this useful product should be given to the farmers, so that the richness, the prosperity goes to the actual person who is doing a lot of labor. And sometimes, unfortunately, in certain parts of India, we have been listening that farmers are committing suicide. So this kind of tendency should be immediately stopped, and government and all the NGOs, the student vice chancellors should open up, should go into this era. And they must think that the actual Annadata is our farmer. And if he's not happy, then what cereals, what anna which we are taking in the form of the food will not be beneficial. मैं कहना चाहता हूं कि किसानों, अगर किसान खुश नहीं है, तो आप ये समझिए उनके द्वारा जो उत्पादन किया जा रहा है अन्न, अगर हम उसका सेवन कर रहे हैं तो हम भी बहुत देर तक खुश नहीं रह सकते. तो ये बात है. मैं कहूंगा कि the actual benefits must be transferred to the farmers to the best. We are ready for certain startups. As many as 20 startups have been opened in our university campus, and some of them are also doing some jobs in the agriculture sector. Some are doing in the biotech sector, some they are doing in the life sciences sector also. So I personally believe that if we use the energy of the youth, which is 65% of our population, then it will be very beneficial for us and for the farmers as well to use this energy. And by the energy of the farmers, by the energy of the students, and by the support of the student, if this synergy clicks, then what the achievements will be greater, and our farmers will be happy. And ultimately, the prosperity of the farmers is very, very essential thing, which I must lay stress on. And therefore, this foundation, FloraFauna Science Foundation, should be given enough credit because it is working in the farmer sector, and it is also collaborating with the research institute like NBRI, like other research CSIR institutes. They are collaborating with the FFI, FloraFauna Science Foundation. So in that way, I feel that some NGOs, some research institutes, some advanced farmers should form a group, and this group should go into the details, what kind of difficulties are there in this farming sector. We are running through an era in which nutraceuticals are gaining a good market in our country. And therefore, if we can raise certain drugs, if we can put the genes into the genome of the plants, we can also produce antibodies. These are edible antibodies. So molecular farming has got its importance in this sector also. Although this technique from lab to land is not reaching at the optimum level, but I feel that in coming few years, with the intervention of many NGOs like you, with the intervention of a great scientist like Dr. Khanuja, this technique should be made available to the farmers, so that we can produce antibodies in the farms, we can produce many nutraceuticals in the farms, and the disease-free India can only be imagined if our farmers are participating in it. So I must thank Dr. Khanuja and his bright team, including our Director of the Agriculture Sciences Department, Dr. Soni Gupta, who are doing excellent job in uplifting the farmers level, farmers economic status, and they are virtually seeing into the prosperity of the farmers. Thank you, sir. Thank you very much.
Thank you so much, sir. Very well said. So to keep our nation happy, we have to keep our farmers happy. Thank you, sir, for the talk. We are very delighted to have the presence of our chief guest here, Professor V.K. Pathak, Vice Chancellor, CSJMU. May I request Dr. Shilpa Kayastha, CSJMU, to please introduce our chief guest and proceed.
Thank you so much. A very good morning to all distinguished participants of the FloraFauna Science Innovation Summit. It is my humble privilege to introduce to you Honorable Vice Chancellor, Chhatrapati Shahuji Maharaj University, Kanpur, Professor Vinay Kumar Pathak ji. A visionary leader, an able administrator, a champion of youth empowerment, Professor Pathak exemplifies the epitome of administrative prowess coupled with profound commitment to nurturing an environment where innovation, creativity, and ingenuity thrive. Professor Pathak did his BTech in Computer Science from Hardcore Butler Technology Institute, Kanpur, and MTech from IIT Kharagpur, and PhD in Computer Science from UP Technical University, Lucknow, under the joint guidance of IIT Kanpur. Research interests of Professor Pathak include computational geometry and image processing. With the beginning of a distinguished academic career as professor in the Department of Computer Science at HBTU Kanpur, Professor Pathak's career is marked by remarkable achievements as a 7-time Vice Chancellor, Uttarakhand Open University Uttarakhand, Vardhaman Mahavir Open University Rajasthan, Rajasthan Technical University Kota, Dr. APJ Abdul Kalam Technical University Lucknow, HBTU Kanpur, Dr. Bhimrao Ambedkar University Agra, additional charge, and presently Vice Chancellor of Chhatrapati Shahuji Maharaj University Kanpur. Recently, he has also taken charge as the Vice President of the Association of Indian Universities. In his tenure as Vice Chancellor, Professor Pathak has consistently demonstrated strategic foresight, hands-on leadership aimed at enhancing teaching and learning methodologies and ensuring quality and holistic education to all. His focus on advancing a culture of innovation has resonated throughout academic communities through strategic initiatives and collaborative partnerships. He has spearheaded efforts to bridge the gap between academia, industry, even farmers as stakeholders, with the establishing of a School of Advanced Agriculture, Science and Technology at our university itself. Under his guidance, interdisciplinary collaborations have flourished, tech business incubators have been established, and entrepreneur, innovation, and startup spirit has been ignited, positioning others as well as our CSGM University at the forefront of innovation-driven education. Professor Pathak has been the chairman of several expert committees and member of many academic and administrative bodies. To name a few, the Task Force Committee for the New Education Policy, National Committee to ensure effective implementation of model curriculum, Board of UP Secondary Education, UP Technical University, and NBA of AICTE. He has several international and national publications, book chapters, patents to his credit, and also the awards of Global E-Learning and the Humanitarian Award. In conclusion, Professor Pathak stands as a beacon of inspiration, a catalyst for positive change, and a driving force for transformative journey of our university. He has elevated our institution to new heights with his foresight. And of course, I'm happy to share that the university has recently been accredited with the NAC A++. As Professor Avasti shared with you, in our university also, we have, under his able guidance, established a tech business incubator with the name of CSGF Innovation Foundation, where we are nurturing and supporting up to 20 startups, of which almost 3 of them are directly in the field of agtech, and others are supporting agriculture-based businesses. Please join me in extending a warm welcome to Professor Pathak, whose remarkable leadership continues to shape the future of our university and inspires generations to come. Thank you so much, sir.
Thank you, Shilpaji, for such a nice illustrative uh, things which you have told for me, which I don't actually deserve a lot. So welcome to this Flora and Fauna conference. Honorable Khanuja Saab, the persons who have given the nice talk, the starting talk, which was very nice. So we belong to a country and we belong to a society where we have said that Uttam Kheti, Madhyam Van, Nikrish Chakri. So agriculture is the best, what we have told years before. So we also belong, this Kanpur University or CSGM University is also very close to the Kavi who has written these kind of things. He is known as Kavi Ghag. And there are a lot of sayings of him, which is very near to us in Kannauj, where he has predicted when to sow, when to ripe, when to do all these things, what agriculture members have to do. So actually, this whole conference was a nice conference in the sense that the speaker, who was the keynote speaker, has actually told us that we have to really do something which is of medicinal plant, and actually the country is changing. I am not an agriculture scientist, but I am very much closely associated with the natural farming, with the organic farming, with the new kind of things where the agriculture people and new innovations, incubations we are seeing, which is happening in the agriculture kind of thing. I think the country has changed, and the concept also is changing in the terms of flora and fauna, what we are trying to do. The only important thing which I have recovered or discovered from this conference is there are new ways to experiment. And as the person who has given the keynote speaker has actually told that ideas, and there are several ideas, there is no algorithm to say you experiment with this or you experiment with that. People should experiment with the new agricultural kind of things, new crossbreeding, a new kind of flora and fauna, new kind of exports, new kind of a marketing chain, new kind of a value-added products, which we can really see. I think our agriculture department is a very new department. They have to learn, and I'm very happy to state on this forum that our agriculture students are also developing a lot of new things. And as I have already told in so many conferences that after the education and after the independence, we have very much compartmentalized the things. Very compartmentalize the things that these biology people will study medical, and the agriculture you need zoology, botany, and to understand and all those things. My feeling is always there that we should open everything for everything. So the new results will also and can come from other people. There is a simple experiment where one of our science student is doing, and they are doing with AI to grow the crops. So it is not only the agriculture people, but the science people, the life science people also have a lot of things to say in the agriculture, as well as in the medicinal plants also. It's not only the Ayurvedic physicians, but the other people also who know about those seeds and about those plants can also have a good kind of a command and can also create a new infrastructure. I think at this moment, I am happy to be part of this such kind of a great conference. Thanks to Professor Khanuja, who has come here also and mentored us. But we have to go a long way. We have to take, and it is the duty of the educational institute or the duty of the people who say that we know something to experiment with the common people to see how the common farmers are working and try to understand and try to grow new things and try to experiment with new things. That is very important, to experiment with new things. We are all already bogged up, or we are always bogged up by the course curricula, other kind of things, which to me, in such kind of diverse field, knowledge is important, the structured knowledge is important, but the unstructured experiment also yields a lot of new things and new ideas and new innovations. So let us prepare our students with structured ideas, with the structured knowledge, already they are doing in the course, but with the unstructured ideas and with the unstructured experiments to perform, they can bring new kind of things. So we should have now new hackathons, if Professor Khanuja will say and start, so that it can be done in a field or some kind of a thing. And if this forum also promotes these kind of things, we can have regular hackathons, we can have regular experiments, those all kind of things we can really do. Thank you very much and wish you a great success for this conference.
Thank you so much, sir. Thank you. Very rightly said that apart from beside structured knowledge, we should also experiment. Experimentation is very important to grow. So I request Professor SPS Khanuja to please present the citation to our chief guest and please sum up the presentation.
Thank you, Dr. Pathak. It has been a great time for us that you joined this summit and also gave you a piece of advice. I'm already associated with CSJMU. Professor Avasthi and yourself have been kind enough to create a niche where innovation can prosper. Soni Gupta, who has been steering this collaboration, also always is active in translating the things. So we look forward to the hackathons you talked about in the area of innovation. And I totally agree with you that experimenting is the way forward. In fact, all my life I have been experimenting with myself and finding that it works. And that's what FloraFauna Science Foundation is doing. And that's what now FloraFauna Science Foundation and CSJMU together would be doing. So let's translate what has been since morning in terms of the mirror that has been shown by Professor Dr. D.B. Narayana, who is a very learned person in the field of pharma, as well as medicinal plants. He has been on National Medicinal Plant Boards and he has been with corporate in herbal industry and so on. So I'm sure the farmers are going to get benefited. And it also gives us an opportunity to have our collaborators who are present today to collaborate with each other. And we will be happy to link them up so that it is not only bipartite, it becomes tripartite, tetrapartite, and so on, so that we keep going to villages and making e-villages in future. Thank you.
So we would like to share the honor. So would you like to
This is
emphasize
a
on this?
small gesture, Professor Pathak. We really welcome you as the distinguished member of FloraFauna Science Foundation, and we recognize the role that you are playing in steering the education sector with the research, science, and technologies, enabling it. And you yourself being a technocrat, we welcome you to this foundation.
Welcome, sir. Thank you, sir. Now, moving towards the last bit of the first session, I request Dr. A.K. Singh, Vice President, FloraFauna Science Foundation, to please present the vote of thanks.
Thank you. We are very grateful to Professor Vinay Kumar Pathak, the Vice Chancellor of CSJMU, for sparing his valuable time to guide us and to deliver the presidential lecture. Thank you, Professor Pathak, and your advice is very valuable to us, to our students, and the budding scientists. Dr. D.B. Narayana has given a very illuminating lecture, and we are very grateful for him for sparing his valuable time to be with us. We have been able to interact with him after a long time, and thank you, sir. We are very grateful to Director NBRI for entering into MOU with FloraFauna Science Foundation, and Dr. Manish Bhoyer, who has represented Director NBRI, and Dr. Neeraj Jain from our side, who have inked the memorandum of understanding 2 days before, which was formally exchanged today in this important Aarambh session of FloraFauna Innovation Summit. We are also grateful to Professor S.K. Avasthi, Pro Vice Chancellor of CSJMU, for his remarks, and he will be here with us the whole day, I suppose, and in other sessions also he will be contributing a lot in blessing us, in guiding us. We are grateful to our founder president, Professor Khanuja, for all the guidance and support we received for organizing this summit. And this is 5th in series. I suppose many more will happen, and some of very important recommendations will emerge out of this summit in the end of the day. Our team of FloraFauna Science Foundation, all the members, and especially I would like to mention few names: Dr. Soni Gupta, who is assistant professor at CSJMU and also an active member of FloraFauna Science Foundation; Dr. Neeraj Jain, our general secretary; Dr. N. Manika, who is scientific program coordinator; and all other members who are present on the panel: Dr. Ram Surop, Dr. Many more, Dr. Anita ji, and all other members who are here, with whom we have been interacting, and without their support, it could have not been possible to organize this event successfully on this scale. And all the participants and all the registrants from different parts of the country, from different institutes, organizations, and our जो किसान भाई हमारे इसमें रजिस्टर किए हैं और जो कि इंटरप्रेन्योरियल सेक्शन में अपना करेंगे, और सभी जो लोग इसमें जुड़े हैं आज आरंभ सेशन में, जो कि इनॉग्रल सेशन है, सभी को धन्यवाद देते हुए मैं आपसे विदा लेता हूं, और आगे की सेशन की कार्रवाई के लिए मैं डॉक्टर मनिका से निवेदन करता हूं।
Thank you, sir. So our next session is Khoj, which Dr. Soni Gupta will lead. And before we begin with the Khoj session, are we taking a break, Dr. Soni, for 5 minutes? Like,
it depends. We were about to, yeah. We'll take a break of 5 minutes. So, sir,
5
as sir
minutes.
says, okay,
Yes.
fine. We'll take a break of 5 minutes and then
Just
gather
5
here.
minutes.
Yeah.
But we need all the Khoj participants to remain here, and so that we can align them here.
You can announce.
After 5.
You can announce the names and those, I mean, who are the first presenters, they should be ready for their presentation.
Yes. All the Khoj
I'll be
presenters, please be here. Dr. Soni Gupta is here to coordinate with you all. And Dr. Soni, how do you like to proceed? Like, you want to call their names?
Yeah, I'll be taking over. I'll be taking over for with the Khoj session along with Dr. Sarika Srivastava. Okay.
Yes. Oh, yeah. Dr. Sarika is here. Please welcome her on board. Yeah.
Thank you, Sarika, for coordinating with me, and she'll be an important aspect handling this session.
Thank you.
We'll just take a break of, we'll just start the session in 4 minutes.
We can continue. We can continue.
Now.
Yeah, I mean, those who want to take a break, they can just put up their microphone. No problem. We can, I mean, we are already late, so we can continue. No problem. Dr. Sarika, you can just explain the rules and all that.
Yeah. I'll do that. So I formally welcome all of you to this session, fourth session. This is basically a session which is the highlight of the summit. Involvement of so many people, be it farmers, be it students, be it entrepreneurs, researchers, and it has always been a very good highlight of the summit, because we have this slot, this fourth session. We go through these e-posters, videos, collages of innovations and ideas. And to sum up, first of all, I'd like to say that we have got around 16 presentations lined up during the session. And these are categorized into 3 major categories, which are basically research, innovation, and entrepreneurship. There are something around 7 presentation categories, 6. First, we have a panel of judges. I welcome all the jury members. Welcome, sir. We have with us Dr. Ashwini Safal, Dr. MP Darukar, and Dr. Shilpa Kayastha. You have been together on a WhatsApp group. We will be going through these presentations online, and the marking on these presentations will be on the basis of 4 aspects. These are content, that is of 10 marks, research, innovation, that's 10 marks, expression, how you are going to deliver.
डॉक्टर सोनी, आपकी आवाज कुछ ब्रेक हो रही है. Dr. Soni.
10 marks.
Soni,
And the significance of
your
work
voice is breaking. Can you be in a network area? I guess you're not getting a proper network.
Just a sec. Just a sec. I'll change the network and.
Sure.
Yeah, sure.
Am I audible now?
अब ठीक बोल रहे हैं, अब ठीक है.
Yeah, thank you. Actually, I changed the network. There was some issues with the network, maybe. So, kind of technical glitch going. Anyways, I just wanted to welcome the jury again. And we have with us Dr. Ashwini Srivastav, Dr. Manisha Singh, Dr. Neeraj Jain, Dr. Ravindra Rana, Dr. Archana Suman, Dr. MP Darukar, and Dr. Shilpa Kayastha. We are all in a associated with the WhatsApp group. There are 4 criteria on which these presentations will be evaluated. Content will carry 10 marks. Research and innovation will carry another 10 marks. We have 10 marks for the expression, that is the delivery of your matter or the content, and the significance of the work presented will again carry 10 marks. We have got 40 marks in total on which it will be evaluated. There'll be the presenters will be given only 5 minutes of time to present their ideas or the innovation or the research work, and it will be followed by 1 or 2 minutes if the judges wanted to ask some questions. So I'll ask Dr. Sarika Srivastav to please start with the presentations, and all the presenters, please. I expect that you have all joined and we'll be starting over with it. Dr. Sarika Srivastav, please start with the first presentation.
Yes, ma'am.
First presentation is of Neha Gosai. Dr. Manisha Singh, are you there, ma'am? Dr. Manisha Singh?
Hello.
Yeah, just to brief the evaluators or the jury members, we have got today in the morning, Manisha ma'am told there was some issue in her city, Haldwani, and Neha Gosai ma'am may not be able to present. For that, I have requested Manisha ma'am to present her work. Please open, Sarika.
Hello.
Yeah.
Hello. I'm audible.
Yes, ma'am, you are audible. Sarika,
Uh,
please open Neha Gosai's presentation, please.
Is it visible?
No.
No, it's not right now.
Yeah, that's true. Yeah.
Yes, it is visible now.
Yeah. If we can request all the jury members to please open their cameras. Or later we can do it for a group photo or something like that.
Yes. Yes.
Okay.
Yes.
Okay.
Hello. I think it
Yes.
is not visible for me. Slide is.
You're not able to see, ma'am. We are able to see the slides.
Okay.
You can start, we can see this and you can see in your computer.
Just a minute, sir. So she is working on the development of biofertilizers. As we all know that we are moving towards the organic farming. So she is just taking the she's collecting the algae, that is the blue-green algae from the different parts of the Uttarakhand. Basically the freshwater streams, and she's collecting in a area where she can grow in a small ponds where we have collected the rainwater, and she's growing that algae along with the crops. And by using that particular algae along with the waste which is from the agriculture field, and she's just incorporating that waste and converting that waste along with the algae to the biofertilizer, which is applied to the crops. Instead of using the chemical, she's just using that algae to grow the plants, and where we can easily, so I don't have just more idea of it, but a little bit idea of her, that she is growing the algae in a field and using that algae, along with she is using that algae to form up plates, that is sheets. As we are growing, we are just supplementing the agar in the form of the agar sheets, similar to the agar sheet, and trying to grow the crops in collaboration or by using that algae as a fertilizer.
Ma'am, can you sum up the conclusions, please?
Ma'am, wait a minute.
Yeah. Can you conclude your slides?
Yes. she is growing the algae along with the waste as an organic fertilizer. So along with, she's also incorporated some of the microorganism along with it. So the plant-based biofertilizer, it will increases the soil fertility along with the microflora. So the productivity of the crops will be increased, and this is one of the I can say this is one of the best practice to increase the soil quality as well as the fertility of it.
Thank you, ma'am. Thank you for this work, presenting this work. We'll move to the next presenter. If the jury members wants to ask a question or two, or else we'll move to the next presenter.
Tony,
No,
let the
really.
jury ask the questions, because that is how we'll be assessing. Okay.
Okay.
Presenter is not here, only good.
Yeah.
That's why it is, I don't think we can ask any question to that.
Okay, fine, sir. So we'll move to the next one. Thank you, sir. The next presenter is N.S. Nysen.
Yes, ma'am.
The next presenter is N.S. Nysen.
Yes, ma'am.
Yeah. We are just opening your presentation. Please be ready with it.
Is it visual?
Not right now.
Devashish. Devashish, present है ना? Present. Yeah, is it visible?
Yes, yes.
Yes, ma'am. Shall I start, ma'am?
Yes, please do so.
Respected scientists and my dear friends, I'm Naishant N.S., PhD scholar, Division of Microbiology, ICR, Indian Agriculture Research Institute, New Delhi. And now I'll be presenting my research work on the topic chickpea nodule endophytic bacteria and their role in plant growth promotion. And chickpea, as we know, one of the most important winter crops and world's third most important food legume with high protein content. And as you all know, these legume crops are capable of forming symbiotic association with different rhizobia. And in chickpea, they form symbiotic association with different species of mesorhizobium. So such symbiotic interaction lead to the formation of a well-differentiated and unique nitrogen-fixing organ called root nodule. And these structures provide specialized microenvironment for the nitrogen fixation process. And inside these nodules, mesorhizobium, they can interact with the host plant and they convert the atmospheric nitrogen into plant usable form. So these structures, they provide a host plant with the direct source of fixed nitrogen, which reduces the host plant reliance on nitrogen from the soil or from chemical fertilizers. So these root nodules are predominantly colonized by rhizobial species. But apart from rhizobia, other bacteria are also present inside the nodules. They are collectively known as non-rhizobial endophytes. And these non-rhizobial beneficial nodule endophytic bacteria, they improve the rhizobial symbiosis, also the nutrient availability and plant growth through multiple mechanisms. But despite their potential role in plant growth promotion, their structure and function remain largely unexplored. And evaluation of plant growth promoting ability of these bacterial endophyte is very important. And there is a possibility of increasing growth and yield of chickpea by using compatible rhizobial and non-rhizobial endophytes. So considering all these points, the aim of the present study was to isolate the bacterial endophytes from chickpea root nodules and to characterize their plant growth promoting traits. And this study was framed with the following objectives. First one is to isolate the root nodule endophytic bacteria associated with differentially nodulating chickpea germplasm lines. And next is to evaluate their plant growth promoting potential. And here you can see the methodologies followed in the study. First, a field experiment was conducted during the rabi season of 2020 to 23 in augmented experimental design. This was to assess the inherent symbiotic potential of 155 chickpea genotypes with a popular genotype BG372, which is released from IRI as a reference check. And at the flower initiation stage, various parameters like nodule number, nodule fresh weight, and nodule dry weight per plant, and also the plant biomass was recorded. And after the identification of low and differentially performing, that is low and high nodulating chickpea genotypes, isolation of the nodule endophytic bacteria was done. And this was done using 4 different standard media, that is yeast extract mannitol agar, Jensen's agar, Burke's modified glucose mannitol agar, and Bund and Malik's agar. And these isolates were then further screened for their PGP, plant growth promoting traits like indole 3 acetic acid production, biofilm formation, phosphate solubilization, and nitrogen fixation. And coming to the results part, first in the field study, here is the frequency. You can see the frequency distribution of 155 genotypes based on the number of nodule per plant. This was taken at the flower initiation stage, and it ranged from 2 to 21 nodule per plant. Whereas the nodule fresh weight, it ranged from 10 to 760 milligram per plant, and the plant biomass, both the shoot and root biomass, ranged from 1.51 to 62.96 gram per plant. And from all these lines, based on their inherent nodulation potential and plant biomass, 6 genotypes, each from high and low nodulating categories, were identified for further evaluation. Ma'am, we can move to next slide. And here the first histogram that represent the inherent nodulation potential, root and shoot biomass of selected chickpea genotypes. And next, from these selected low and high nodulating chickpea, a total of 33 rhizobial and 36 non-rhizobial bacteria were isolated. And these rhizobial and non-rhizobial isolates were further screened for their PGP, plant growth promoting traits. And coming to the rhizobial isolates, first, the indole 3 acetic acid production. Here you can see in the first histogram, the indole 3 acetic acid production. This ranged from 1.55 to 6.53 microgram per liter of the culture filtrate. And in case of biofilm assay, the mean absorbance at 550 nanometer, that ranged from 0.01 to 1.28. And out of 33 rhizobial isolates, only 3 isolates, they showed positive reaction in qualitative assay for phosphate solubilization. And coming to the non-rhizobial isolates, the same screening procedure was done for all the 36 non-rhizobial isolates. And here the IEA production ranges from 1.13 to 5.82 microgram per liter of culture filtrate. And the biofilm formation was from 1.13 to 0.992 OD. And when checked for the phosphate solubilizing activity, none of the non-rhizobial isolates showed phosphate solubilizing activity. And have done a qualitative assay for nitrogen fixing ability also for all these non-rhizobial isolates using nitrogen-free bromothymol NFP medium. And here the positive reaction was indicated by a color change from yellow to greenish blue. And the 19 non-rhizobial isolates, they exhibited positive reaction. And actually, this indicates their ability to fix nitrogen. And additionally, 13 isolates exhibited pellicle formation in semi-solid nitrogen-free bromothymol blue medium. And coming to the important findings of my study, there was a wide variation observed in inherent nodulation potential and plant biomass among different chickpea genotypes. And apart from mesorhizobium, this study could confirm the presence of many non-rhizobial endophytic bacteria in chickpea root nodules. And all these non-rhizobial endophytic bacteria, they showed diverse functional potential like IAA production, biofilm formation, phosphate solubilization, and nitrogen fixation. And coming to the conclusion, here I'm concluding my study. That is, this study elucidates the symbiotic performance of differentially nodulating chickpea germplasm lines. Also, the study highlights the existence of non-rhizobial nodule endophytic bacteria in chickpea, but their structural and functional diversity needs to be studied in detail. Moreover, a comprehensive validations are also required to explore the full potential of non-rhizobial nodule endophytic bacteria in chickpea, also along with their synergistic role in symbiosis. Thank you.
Thank you. Thank you, Niasid. And the jury can please ask questions related to this presentation.
Such work has also been done previously. No, I would like to know what is the most significant point which you have observed. And because such work has also been done earlier also.
So actually in chickpea, this non-rhizobial endophytes have been explored very less. Actually, these non-rhizobial endophytes, they are actively they help in rhizobium present in the nodule for the effective symbiosis. So if we go for only the rhizobial, if we focus more on the rhizobial isolates only, and if we combine this compatible rhizobial and non-rhizobial isolates in future, they can improve the efficiency of nitrogen fixation as well as plant growth promotion.
Okay.
I would like to just know one point that you
Yes.
have taken the observation of plant performance at pre-flower or at flower initiation stage.
Yes, sir. Yes, sir.
What was the point in that? Because in chickpea, ultimately the fruit would have mattered, or how much was the fruit yield, the chickpea yield?
So
Why
this
did
is
you?
the state.
Why
This
did
is the.
so?
Sorry, sir.
Yes.
Hello.
Hello. Yes. Yes.
Nicen's question is that why you took this at flowering stage?
Sir, okay, sir. Sir, this is the stage at which maximum nodulation we can observe. We are more focusing on nodulation potential rather than plant by or final yield and all. Because if the nodulation potential is high, means that is directly correlated with the plant growth and its yield. So here we are focused on plant nodule dry weight and nodule fresh weight and nodule number as well, sir.
Okay. Okay. And do you think this is reflective of the final fruit formation? Can we say that this is our studies you have done? Can you say with some degree of confidence that this growth performance can be reflective of that yield also?
Yes, sir, definitely. Yes, sir. Because this nodulation is directly correlated with the nitrogen fixation process. And if the nitrogen fixation efficiency is high, means that will help in plant growth and ultimately in yield. It will reflect in yield, sir.
Okay.
Hello, Nysa.
Yes.
Can I ask question?
Yes, sir, sure.
I want to ask two questions from you. That what is your thought process that why this non-rhizobial isolates are present in nodule? Till now, we were considering that a nodule is means a certain house of rhizobium only.
Yes, ma'am.
Yeah, and the second, first you answer this, then I'll ask second question.
Ma'am, apart from rhizobia, there are many other bacteria which is directly or indirectly involved in nitrogen fixation process. Because this rhizobia alone, rhizobia, they are involved in nitrogen fixation process. But when we consider other plant growth promoting traits, they may or may not be involved in other plant growth maybe possessing other plant growth promoting traits like IAA production or cytokinin, gibberellic acid, all other plant growth promoting traits. So these bacteria, other bacteria residing with the rhizobia, they contribute more towards other plant growth promoting traits. So they can improve the nitrogen fixation process. So for example, they can, by producing IAA, that will contribute to plant auxin spool, and that will results in more root development. That is directly linked to the nodule development.
Yes.
So it is directly or indirectly involved in nodule formation as well.
Correct. Because endophytes, it is a common behavior of a plant to pick up, to recruit few microorganisms. So when they are recruited in case of pulses, it is helping the nodulation process or helping the nodule formation much more.
Yes.
Correct?
Yes, ma'am. Yes.
And one more thing which you should, you gave biofilm in the form of OD. You have given OD. Convert it into a quantitative data.
Okay, ma'am. Okay.
Make
Sure.
standard curve with either some dye which is used to check the biofilm. Which dye you used?
It is crystal violet, ma'am.
Crystal violet. So you can make a standard curve with the crystal. There is a methodology, and you should convert this data from OD to quantifiable terms.
Yeah, sure, sure.
Anyway, good work. Good luck. Good luck. Thank you.
Thank you.
Thank you, Naisa. And as ma'am said, good work, detailed work. So keep your fingers crossed, and there'll be other presenters also.
Dr. Soni, I would like to interrupt a little. Sorry for the interruption. Do you have Devashir's slides?
Yes, I have. I've just informed him.
Okay.
He's
Great.
he's under the innovation theme. Okay.
Okay. Great.
We are going through the research theme right now.
Okay. He had some confusion, I suppose. He
No,
messaged me.
I've just sorted it out.
Okay, thank you so much. You can continue.
Sarika, can we move to the next presenter? The next presentation is by Ashwini Sharma. He's an MSc student. Can we have his presentation, please, Sarika? Ashwini Sharma, are you ready there?
Yes, ma'am.
Yeah, sure. Please go ahead.
Good afternoon, everyone. I am Ashwini Sharma, currently pursuing MSc Biotechnology from CSGM University, Department of Biotechnology. I studied study of paddy cultivation, entitled as Assessment of Paddy cultivation impacts on greenhouse gas emission, water usage, and sustainable agriculture practices in Assam, India. Next slide, please. Yeah. I have studied the Assam area, as it is holding 9th position in the country, producing as approx 50 lakh tons of rice annually and contributing about 4.2% of national rice production. This my objective is to assess the annual water demand associated with greenhouse gas emission and water consumption by paddy in Assam. To conduct this study, I have collected different sources of data like IPCC report, historical yield information of Assam, and agriculture data of Assam, and also the economic survey of Assam. According to the IPCC reports, the approx 71.32 million metric ton of GHG emitted from India. After this study, I have been considered as approx 3.7 billion liters of water utilized by paddy to cultivate the paddy in Assam, which is equivalent to the 5 years water demand of city like Delhi. And it also contributes approx 4.7 million metric ton of GHG emitted from paddy fields annually in Assam. The conclusion of this study is that approx by emitting 0.9 kg of carbon dioxide uh, uh, in the production of a kg of rice, it contributes approx 4.7 million metric tons of GHG, and approx 2,000 liters of water utilized to grow 1 kg of rice. The water required to grow paddy in Assam is approx 3.7 million liters of paddy cultivation in Assam, which is equivalent to 5 years of water demand for a city like Delhi. As the 70% of Assam's population relies on agriculture, judicious crop planning and management, especially in vulnerable blocks, will play a crucial role in climate resilient and smart agriculture practices for ensuring the sustainability of agriculture and achieving net zero emission by 2070. Thank you.
Thank you, Ashwini Sharma. The jury can now ask questions. He's basically done this survey in the state of Assam on paddy cultivation.
Shall I ask first?
Yeah, please, ma'am, please.
Okay, Ashwani. Good attempt. You are surveying, it's sort of a survey which you have done. When you surveyed, what was the position in Assam, or you know anything about the alternative to this for cultivation of rice? That what the government or scientific community is advocating? Ashwani? Ashwani is the name, no?
Yeah, yeah.
Yes, ma'am.
Hello, Ashwani. Could you understand my question? You know, rice cultivation, a lot of water is needed. That's why you have given such a high water footprint for 1 kg of rice, around 2000 to 2500 liter of water. What is the alternate technology of cultivation of rice? Because we can't live without rice, especially the area which you have
Yes, ma'am.
selected or the southern part. Then what is the alternative?
Basically the north is the more capable of growing rice, but it also leads to GHG emission. GHG emission.
Yeah.
The alternatives of these may be like millets crops and some other crops maybe.
And if you want to eat rice only, then if you do not want to shift to the some other grain, if you want only rice, then is there any technology which is coming up in a big way? Do you have heard DSR? Direct seeded rice.
Yes, ma'am.
Anna? So you should in your survey you should see also that technology is coming in a big way. Where it is a direct seeded rice. So you should see that how much requirement is there in that. And another thing, you are talking of methane emission. So you should know why this methane is emitted from rice fields.
Ma'am, methane emission basically due to the flooded irrigation, because the anaerobic bacteria present in the paddy fields will lead to anaerobic respiration, which ultimately leads to a GHG emission.
Very
And
nice.
various the use of lot of fertilizers will also lead to GHG emission.
Good. Okay.
Good. Yes, thank you.
Any other questions from the jury members?
No, it's
Hello? Hello?
okay.
How many crops do you grow in Assam area for rice?
Yes, ma'am.
How many crops? How many
Hello.
crops of the rice will be cultivated in Assam area?
Cycle of crops, madam is asking.
Yes.
Basically, in Assam, approx 2 or 3 cycles, people farmers of Assam cultivate 2 or 3 times in a year of paddy.
All will be needed of water continuously.
Yes, ma'am. The paddy requires continuously water to grow.
Instead of using this mulch cropping in the rice, we can grow for the direct seeding. That will be beneficial, or it can save the water.
Yes, ma'am. It is. Hello.
Yes, please go ahead, Ashwini.
Ma'am, can you please ask again?
Ma'am was asking the direct seeded rice varieties which can be grown. Are you saying yes to that? your presentation? Adapting new technologies. You are
Yes, ma'am.
basically
Adapting new
giving a data of rice cultivation in Assam, where the water footprint or the greenhouse gases emission is quite high. It is a good data that you have collected, and it is high time we sit together and think what kind of newer technologies can be adopted. We should move towards climate resilient agriculture, adapting new other crops to integrate in our food. And if we are going to stick to rice, then we have to go in with newer technologies which can be of sustainable nature. Is that what
Yes,
you're
ma'am.
saying, trying to say? Okay. Okay. Thank you, Ashwini. Anything else you like to add to it?
No, ma'am. Thank you.
Okay. Thank you, Ashwini. Can we move to the next presentation, jury members?
Yes, you can.
Please, Sarika, can you please open the presentation of Ms. Avantika Siddharth?
Is it visible?
Not yet.
No, ma'am. Now?
No.
Now is it visible? Here is.
No, it's not come yet.
Oh, wait, sir.
Yes, please move to the first slide. Yeah. Avantika, please start your presentation.
Ma'am, slide is not visible.
No, it is visible at my end.
Yeah, is it visible?
Avantika, unmute yourself and please start your presentation.
Ma'am, slide is not visible at my end.
Okay.
Reshare it then.
Yeah, sure, sir.
Sarika, please let me share her screen. You please stop the sharing, I'll share it. Visible?
Yes, ma'am. Now it's Now it's visible.
Please go ahead. Please go ahead.
Good afternoon, everyone. Myself, Avantika Siddharth. I am presenting on the topic estimating rainwater harvesting potential using GIS technology. Rainwater. Ma'am, can we move to the next slide? Rainwater harvesting is the local collection. Rainwater harvesting is the local collection and storage of rainfall through different technologies for future use to meet the demands of human consumption or human activities. In our Earth comprises 29% land and 71% of water. In 71% of water, 96.5% is oceans, 0.9% is saline water, and 2.9% is fresh water. As we know, 68.7% of fresh water is in glaciers and ice caps. 1.2% is surface water and 30.1% is groundwater. We depend upon the service of ecosystem to the health way of maintain our health, way of life, and economies. Restoration of rainwater harvest. Rainwater harvesting improves human health without impairing water's environment service in the local environment. As we know, to calculate, so there are two objectives: to calculate area of CSGM University using GIS and estimating rainwater that can be harvesting. To calculate, harvest the rainwater, we we have to calculate the area around in which the optimum rainfall is. There are some methods to calculate the area like GIS technology, and we have the formula to estimated the harvested rainwater. Q is equal to A into C into P. Q is the amount of rainwater that can be harvested. A is the area that receives rainfall directly. C is the runoff coefficient, and P the average annual rainfall. Methods, we have to decide the location where rainfall is conducting, and second, estimation of catchment area, third, measurement of average annual rainfall कि annually कितना rainfall है, third, calculation of runoff coefficient, fourth, calculation of rainwater harvesting potential, and third, determination of water quality that can be harvested from rainwater. As a result, the total area of the university is approx 25 to 259.46 acres. Ma'am, can we move to the next slide? There are some finding and conclusions. Type of area calculated, and runoff coefficient of the different types of lands: country, vegetation land, and open land. To in as our Indian government is also started a campaign, Jal Shakti Abhiyan, Catch the Rain, on March 22, 22nd March and 2021, with the theme Catch the Rain, Where It Falls and When It Falls, is to encourage the states and other stakeholders to build rain
Cannot hear you.
Avantika, you able to hear me? I guess she's having network issues. Maybe she's not assessing the network from the university. That is why. Avantika, are you able to hear?
Tell her to get a better network. In the meantime, we can move to the next.
Yeah, move to the
We
next
will bring her
presentation.
again.
Yeah. We have the next presentation lined up of Sangram Gharai. I'll just open this.
Yes, ma'am. Am I audible?
Yeah, you are audible. Just let me share.
Can I share?
Yeah, if you have, please share it. I have. Wait. Sangram, can you start with your presentation?
Yes, ma'am. Good afternoon to everyone. Myself, Sangram Garai.
a PhD first year student at IRI New Delhi. And I am here to present my topic, which is design and optimization of acid pretreatment parameters for maximum reduction sugar from biomass using RSM. Next slide, please. So we know that India produces around 320 million ton biomass waste each year. So this may create problem like some lack of burning, like rice straw burning. So we need a sustainable technology where we can use and we can gain profit from that biomass. So my research objective was to optimize acid pretreatment parameters, which are pH, temperature, and loading for maximum recovery of reduced sugar from biomass. So this sugar can be utilized for further various products, like as biomass is rich in hemicellulose, cellulose, and lignin. So mainly here I have targeted hemicellulose and cellulose. As we know, cellulose is rich in glucose, and hemicellulose it is rich in ibinose and xylose and other sugars. So here what I have done, here we have taken three variables, like a factor 1, which is temperature. We have taken three level. One is 100 degrees Celsius, 110 degrees Celsius, and 120 degrees Celsius. And another variable, which is loading, weight by volume. There we have taken 10% of biomass, and another is 15%, another is 20%. And each we have treated with different acid concentration level with H2SO4, which is 0.5% and 1% and 2%. So we have conducted our experiment in 15 aqueous tubes in small scale using box Buchanan design, using design expert software. There we have checked for two parameters. One is concentration of the reduced sugar in the hydrolyzed and the total sugar recovered. So the obtained data was analyzed using denser reagent, and the data was analyzed using multiple regression analysis to fit a linear polynomial model and the two-factor interaction model to compare response surface. The coefficient of the model were estimated using ANOVA and determine the significance of each factor and their interaction. And the final model was validated using the software. Then residual analysis was performed to validate the model and using lack of fit test. Next slide, please. Coming to the result, so after feeding the result to our software, we got two responses. So the first response, linear response, was you can say minus 423.20 plus 4.15 into factor A, which is our temperature, and then 1 plus 12.62 into B, which is factor B, and 59.62 into C, which is factor C. So another two-factor interaction we got, and that response was 15.41 plus 0.36 into factor A minus 5.26 into factor B plus 30.22 into factor C, and their combinations 0.05 into factor interaction of factor A and factor B minus 0.45 into interaction of factor A and factor C, and plus 1.82 into interaction of B and C, and each solution was plotted in control plots and the response was found that at a temperature of 120 degrees Celsius, at 2% acid concentration and 20% substrate loading, it is giving maximum sugar concentration as well as maximum volume sugar recovery from the biomass, which can be further used. Like the sugar glucose we are getting, we can produce ethanol for the ethanol, which can be utilized for the blending of blending with fuels, and we can get from xylose we can get xylitol, and from arabinose we can also get different value added products which are of industrial importance as well as medical importance. Thank you.
Any questions from the
Before we
mentors?
go into questions, an advice for you, Sangram. In your title, you are saying RSM.
Yes.
No one knows what it is, and you never explained in the whole presentation. So always avoid using abbreviations in the title, and if at all you have used, your first line in your presentation should be what is RSM. Okay, please do that and then go ahead.
Sangram, I did not understand one point. What is 20% substrate loading? How did you calculate?
20% substrate loading?
Yes, what's that?
Like, sir, in if we are taking 10 ml of acid solution, and we are putting 1 gram of substrate, that is biomass, so it is 10%. So we have taken gram and liter.
Okay. So this is how you calculated it.
Yes, sir.
Okay. And can you provide us some relative cost benefit analysis of this method for getting these reducing sugars from the biomass rather than the waste biomass basically you're talking about? Any idea about that?
Sir, I didn't do that one, cost benefit analysis, but definitely it is a like easy solution, because price of acid is less compared to other treatment which is available for production of sugar from biomass.
And what was the
like, sir, enzymatic activity is it is costly. Yes. Yes.
What was the inspiration for you to take up this kind of a study? Why did you take up this study? Certainly some thought process must have gone. Thought process must have gone.
Yes, sir. Yes, sir. Yes, sir. That as India, government of India is also promoting blending of ethanol in fuel, and also there is a burning problem, that like rice straw burning. So this two thing can be there is one solution which can mitigate these two challenges. So we can use that straws and biomass, and we can produce ethanol.
Okay.
Not only ethanol, Sangram, it is now waste is being used for many products. All chemicals, biochemicals, they are produced by using these waste products, no? So first thing, you have given in result your RSM form, but you should have given in that response surface model, no, where you standardize various options, that how much each substrate should be used, or each part should be used to give the maximum yield, isn't it? You have written in result, but suggestion is very right, that because you are dealing with waste, so you feel that everybody knows that, but it is a very good statistical tool which has come up, which gives you indication that what will be the final thing which will give you the maximum yield. That is, I think, the objective. isn't it?
Yes, ma'am.
Yeah.
Yes, ma'am. Yes, ma'am.
And an idea about how much of waste is getting generated annually or in a particular period, and then how much of ethanol or some valuable product can we get? What is the potential of using this technology for the waste for this purpose?
Yes, sir. In each year,
Impact to your presentation.
You should have included in the introductory part, which you were speaking, but it should have been listed somewhere also, because this is and the substrate loading, because if we take 100% waste, then it is not hydrolyzed, it is not converted into sugars. So we have to grade it, that at which level substrate should be taken up to give the maximum yield. So where he was standardizing the substrate loading, and it is a very essential part when we do some production of industrially important compounds. So you, Sangram, you are a very good student, so you should explain all these things. Okay?
yes, ma'am.
But nice.
I just had one query. Actually, this is something has come out also regarding my query, that is this substrate specific? What kind of substrate have you used, and are you standardizing for different substrates also?
Yes, ma'am. I have used specifically corn cob. Corn cobs. Actually, in whole corn, it counts about 20 to 30% that corn cob waste is generated. So per kg, we can guess around 200 to 300 gram corn cobs will be generated. That I have used as a substrate.
And do you plan to use it for standardize it for different substrates also? Do you want to study that also?
Yes, ma'am. It can be because for different substrate it will be
Yes, because Sangram,
different
you
composition.
link it with the means in any ways you are interested in what? How much lignin is there? How much cellulosic part is there? How much is poly in this hemicellulose is there, na? So these conditions, if you are taking corn cob, more starches will be there. So technology will become slightly different. If you are taking paddy straw, it will be slightly different. If you'll take some woody material, you will have to remove first the lignin, then start this process. So that will depend upon which raw material you take. I think Dr. Neeraj, that is the, I think, basis, because the whatever is
Yes,
the complex carbon will make the initial statement for deciding about the technology.
Yeah, actually, that will be like the more wider use of the technology, then we can do that. With these things are also taken up. Yeah, thank you, Sangram.
Yes, ma'am. Okay,
Hello,
thank you.
Sangram, there is one question.
Yes, ma'am. Yes, ma'am.
Why are you using only the acid? Can we use the bioenzymes instead of these acids?
What can you repeat, ma'am, please?
Ma'am, why are you using the enzyme acids? Instead of using the acid, we can use the bioenzymes for producing the reducing sugar or increasing the content of it.
Ma'am, first enzymes they require time, and crude enzyme is like bioenzyme production. It is also a difficult task, and it will produce in very low amount, and our biomass is huge. So we need this acid treatment is very fast. Means within 20 minutes or 30 minutes we can get sugar. And it is also cheap, and for production of bioenzyme we have to that amount of bioenzyme would need time, as well as it is costly. It will add this means in terms of cost benefit ratio, it will be like very high.
If you are using the this acid, it is drained off in the water. It is creating the water pollution. How it can be environment safe?
Ma'am, after this we generally dilute it so that fungus can grow. Like after this, what we use means what I have done is try to production of try to produce ethanol as well as xylitol. So fungus requires generally a pH of 5. So it is already probiotic that environment, and we have diluted it so that fungus can grow. After that, we have to again, like as you are saying, that we have to drain it or something. But we are neutralizing it with alkaline.
Okay, then it's good. Then it is a good.
Okay. Thank you.
Call next one.
Yeah, thank you, Sangram. We'll be moving to the next presentation, and we have the next presenter, Kavya T. I'll just share the screen. Kavya,
Yes, ma'am. I'm audible now?
please, your voice is very low.
Now I am audible now?
Yes, yes, yes. Please go ahead.
Good afternoon to everyone. Myself Kavya, PhD student from the Division of Microbiology, IRI, New Delhi. Today I am going to present my poster on the topic that the exploring the plant growth promotion potential of the actinobacteria for inducing salt stress tolerance in rice. Next slide. Coming to the introduction, as we know, is the salinity is the most important limiting factor for the cultivation of many agriculture crops like rice, wheat, and barley. And the tolerance level of these cereal crops to the salinity varies from the barley being the most sensitive, and the rice is also one most sensitive crop among the cereals. Keeping in this mind, recently the plant growth promoting rhizobacteria have attracted the attention of many researchers as a promising approach to enhance the plant growth and crop yield under the stressful environmental condition. So in this study, we are assessing the role of the actinobacteria, which belong to the gram-positive group of bacteria having high G plus C content in that cells to mitigate the negative effects of the salt stress in the rice crops. And coming to the material methods, for this study, I have selected the 40 actinobacterial cultures, 27 from the rice rhizosphere and the 13 from the endosphere of the rice, which are already available in our lab. And I have screened these isolates for their morphological characterization based on their pigment production, colony nature, and their spore formation using the 6 different actinobacteria specific media like HPDA, ISP2, ISP3, ISP4, and ISP5 and ISP7. And these actinobacteria were also screened for the different PGP traits using the standard protocols, and also physiological stress related parameters like membrane stability index, chlorophyll content, pollen content, and antioxidant activity were also analyzed using the standard protocols. And also I have evaluated the PGP potential of these selected stress tolerant actinobacteria isolates for rice seed germination and growth under 3 different conditions. One by using in vitro condition using Petri plates, and one by using growth tube experiments, and also by pot experiment. Coming to the results, after the selecting the cultures from the laboratory, I have screened these cultures for their morphological characterization using different medias, and all the isolates were shown they are different to each other based on their morphological characters. And after their morphological characterization, I have screened these isolates for their different tolerance level to the different concentration of NaCl, ranging from 5%, 7.5%, 10%, and 12.5%, and up to 15%. And all the isolates, don't change. All the isolates were most of the isolates were able to show their growth at 5%, 10%, and 7.5% percentage of the NaCl concentration, but very few were able to show their growth at 12.5 and 15% NaCl concentration. Based on their NaCl tolerance level and PGP traits, I have selected the 3 rhizospheric actinobacteria isolates and the 3 endospheric actinobacteria isolates for the pot experiment and growth in vitro experiments. And after the selection, I have also molecularly identified by isolating the DNA and PCR amplification of 16S RRA gene sequencing. The sequencing results shown that all the isolates, most of the isolates were belong to the Streptomyces species, except the one that is MET24, which was identified as a Cytospora species. Next slide. After their molecular identification, I have evaluated the PGP potential of selected stress-tolerant actinobacteria isolates for rice seed germination, seed vigor under a salt stress condition. For this experiment, I have selected the salt-susceptible variety of the rice, that is Varadhan, and salt-tolerant variety, that is BPT, as a control. And all the rice seeds which were treated with the stress-tolerant actinobacterial isolates were after 10 to 15 days, were able to show significant improvement in the shoot length, root length, seedling dry weight, seedling vigor, index, percent germination, and energy of germination compared to the uninoculated controls and also rice tolerant variety BPT. And also, I have evaluated the effect of the selected stress-tolerant actinobacter isolates under the pot experiment, both with in stress and without stress. The results shown that there is a significant improvement in the total chlorophyll content, proline content, and cell membrane stability, and total antioxidant activity of all the plants which were treated with the selected stress actinobacterial isolates compared to the uninoculated control, both with stress and without stress condition. So from this study, I can conclude that by seed and seedling method of inoculation of the stress-tolerant actinobacterial cultures, which were isolated from the rhizosphere and endorhizosphere of the rice crop, has significantly improved the rice growth and also it induced the plant growth promotion traits in the plants, and also improved the physiological adaptive responsive traits during the salt stress condition in the field. Thank you.
So I'll invite questions from the jury.
Hello. Hello.
Hello.
Kavya, you have studied that there is a condition created for the stress condition. There is some stress condition created in this plant, that is typically the salt. Have you studied the correlation between these factors?
No, ma'am. I just different concentration of NaCl I have put in the media, and I screened the cultures.
Relation between the chlorophyll content and the proline content means both they are directly correlated.
No, ma'am. I didn't did any correlation study, ma'am. Separately, I have conducted it.
No, Kavya. You have chlorophyll data and you have proline data, you have yield data. You can always correlate, no? You have not done it, not shown it here. But simple correlation, even if you have not done, do it.
Okay.
Both the data are there, yield parameters are there. Simply it's a calculation, then you can correlate it.
Okay, ma'am, I will do.
Because then only it will show that if proline or if these conditions are modulating the plant for staying green, or chlorophyll content is there or not, how much it is dealing. You have shown your plants. They are showing green in salt stress or not?
Ma'am, they are showing green, ma'am. Some cultures were green, some cultures are not green.
Yeah, that is what madam is asking. So, but you in your this to finish it, you correlate also.
Okay, ma'am, I will do.
My question to you, Kavya, is why you took actinobacteria? Why not normal gram negative? Is there any advantage of taking actinobacteria? Because nowadays it is becoming very popular.
Yes, ma'am. Mostly the actinobacteria research by concentrating on that they are very efficient in secondary metabolite production. So nowadays they are concentrating. They are producing spores also, and many PGP traits also they are producing.
Yeah.
So
with spore, what is the advantage? What is the advantage having any structure, any culture producing these resting structures, spores? What advantage it will show?
It can able to tolerate adverse climatic conditions.
Yeah. So long-term survival. So that as a microbiologist, you should answer it very quickly, no? So
Yes, ma'am.
more resilient, more effective colonization, more response.
And they'll produce exopolysaccharide soil aggregation, which will help to hold the water. In some experiment also, they run their role in drought stress condition, ma'am.
Yeah.
Yeah.
So that's why actinobacteria is becoming very, very popular nowadays. No?
Yes, ma'am.
Any commercial formulation of actinobacteria that you know, which is already in the market?
No, ma'am, I don't have idea.
So search that also, because
Okay.
you should, being a worker of actinobacteria, you should know, because in market so many products are there for bacillus, for other bacteria, but this also you should know.
Okay, ma'am.
Thank you. Good work.
Thank you.
Thank you, Kavya.
Thank you.
We'll move to the next presentation. We have Smriti Sachan from CSJMU University. Smriti, please go ahead.
Good afternoon, everyone. Am I audible?
Yes, you are.
Yes.
Good afternoon, everyone. My name is Smriti Sachan, a PhD scholar at Department of Life Sciences, CSGM Kanpur. Today, I am presenting on the topic genome editing technologies for crop improvement. Next slide, please. Genome editing is a relevant, versatile, and preferred tool for crop improvement, as well as for functional genomics. The advances in gene editing techniques, such as zinc finger nucleases, transcription activator like effectors, nucleases, talents, and clustered regularly interspaced short palindromic repeats, CRISPR, associated with the Cas9. These tools supports great opportunities for the future development of plant science and rapid remodeling of crops. Genome editing is a cutting edge technology that allows scientists to make precise modification to the DNA of plants. These modifications can lead to improvement in crop yield, disease resistance, and nutritional value. Zinc finger nucleases. Zinc finger nucleases revolutionized the field of genome manipulation studies as the initial actually aimed protein reagents and are DNA binding components that can detect 3 base pairs at a target site. Different kind of plants including Nicotina tabacum, Arabidopsis thaliana, and maize have all seen widespread use of ZNF for intended genetic modifications. Zinc finger nucleases are one of the among the most effective and efficient tool for modifying gene by focusing on double strand breaks. Using genetically modified nucleases, an initial version of genome editing methods based on zinc finger nucleases was created. Essentially, zinc finger nucleases structural makeup consists of 2 domains. First, the DNA binding domain, which has 300 to 600 repeats of the zinc finger. The range of base pair that each zinc finger repeat can monitor and read is between 9 to 10. And second, the DNA cleavage domain, also known as the specific cleavage domain of type 2 restriction endonuclease. Fork 1 serve as the DNA cleavage domain in zinc finger nucleases. Um, uh, Zinc finger nucleases are composed of 2 monomers. Their respective target sequence reversely flanking the DNA target by 5 to 6 base pair. DNA's flanking sequence is cut by dimers with fork 1 domains and a zinc finger domain with a specific or uncommon targeting site in the genome keeps track of the particular sequence of 24 30 base pair. The capability to modify and influence both advanced and fundamental genomic target is advancing the field of genome editing. Now, transcriptional activator like effector nucleases, talents. A different site driven mutagenesis genome editing system called talents which shares a similar idea to ZNF, was first identified in plant pathogenic bacteria Xanthomonas, and talents are more precise because they only target one nucleotide at the target site instead of three. Genome modification with talent has been effective for angiosperms and bryophytes. The talent system, which has been in use for several years, is a widely used technique for precise genome editing. The fork1 cleavage domain and the binding domain of tail proteins were combined to create talents. And tails are made up of 34 amino acid multiplex repeats that efficiently use one base pair. Similar to zinc finger nucleases, talents also encourage targeted double-strand breaks that support the initiation of DNA damage related pathways and guarantee modifications. The protein engaged in the talent mechanism have a nuclear localization sequence and a central domain that binds to DNA for the first time. It was discovered in 2007 that these proteins have the ability to bind to DNA. In overall, DNA repeat sequence can influence tail proteins by binding to them. Previous research demonstrated that tail proteins always functions at the 50-50 and thymidine base to fix the nucleotide of the DNA sequence. The activities of tail recombinase and tail transcription factors are diminished in the absence of 50 and thymidine base. Since the modulation they provide is much easier, more cost effective, and has a much lower target effect than zinc finger nucleases, talent have become popular over zinc finger nucleases. Now, clustered regularly interspaced short palindromic repeats, CRISPR-Cas9, one of the most popular genome editing tools. CRISPR-Cas9 enables researchers to make precise alteration to crops DNA, such as introduction of advantageous traits or the elimination of detrimental traits. And the use of this technology has the capacity to significantly enhance plant health, overall crop yields, nutrient content, disease and pest resistance, tolerance to environmental stressors, and resistance to pests. Researchers can now successfully knock out, replace, and insert genes with high accuracy and efficiency thanks to genome editing tools. In order to precisely alter target genes in plant, that cutting-edge genome editing technique, CRISPR-Cas9 make use of bacterial immune system. It is made up of primarily of the Cas9 enzyme and the CRISPR RNA. The enzyme Cas9 enzyme functions as a molecular scissors, cutting the target DNA at the predetermined location, while the CRISPR RNA is made to match a specific DNA sequence in the plant genome that needs to be modified. Transfection of plant cells with CRISPR-Cas9. Several techniques are used to introduce the CRISPR-Cas9 system into plant cells, including agrobacterium-mediated transformation, biolistic particle bombardment, protoplast transformation, and viral vector. With the aid of these delivery techniques, the CRISPR-Cas9 system can efficiently and effectively introduce genetic alterations into plant cells, resulting in the desired trait improvement in crops. Gene knockout, replacement, and insertion can be accomplished quickly and effectively with the CRISPR-Cas9 system. This makes it possible for scientists to examine how particular gene in plants function and comprehend their part in various biological processes. The precision plant breeding field has also found CRISPR-Cas9 to be useful tool. Plant breeders can speed up the process of crop improvement by introducing desired traits into plants by manipulating and controlling target genes. Additionally, the desired trait to these modified plants have improved, including disease resistance, increased yield, and increased nutritional value. The field of plant biotechnology has been completely transformed by CRISPR-Cas9 tool for plant genome editing. Plant genome editing has never been easier thanks to the CRISPR-Cas9 technology, simplicity and adaptability and high accuracy. Now, applications in agriculture, various applications in agriculture, as I said, crop improvement, by modification. And now final conclusions. Scientifically speaking, it makes no sense to regulate processes that result in a single extremely precise genome change while leaving unregulated processes like chemical and radiation mutagenesis, which result in thousands of random mutations. We believe that CRISPR and other gene editing technologies have already made significant contribution to crop breeding, but that this is just the beginning of many exciting new developments. Thank you.
Thank you, Smriti. Can you please tell the jury what kind of genome editing technology are you applying on which crop? If you are talking on this technology, are you doing such kind of experiment?
Yes, ma'am. I am doing a genome editing in chickpea for yield attributing traits. So I'm using CRISPR-Cas9 technology for developing a new crop variety by introducing small modification in a yield-related trait.
Any questions from the jury?
Which trait? What's the name? Smriti. Which trait in chickpea you are targeting?
Yield attributing traits like a GM JAG.
Tell the
I'm
trait
targeting
associated with that gene.
G associated with yield. Number of seeds per pod increment.
Yeah.
Okay. So since you're working in some crops, so you do you know any successful study in crops which is already getting commercial very soon? Which has already been under trials and it will be released soon. Do you know any and do you have any information of any success case?
No, ma'am, I don't have any idea.
No, you read about it, no? Because rice is now drought resistant rice, it is coming. It is already in, it has gone into All India Coordinated Research Trials. It is from Indian Institute of Rice Research, Hyderabad, and one from IRI, and they are already into trial, rice especially. Okay.
Okay, ma'am. Yes,
Please
ma'am.
go through this. Ma'am has given a crucial information.
You
Yes,
read
ma'am.
about that. It is already, because if you are working with chickpea, you know that any variety to be released in India, it has to go under some All India coordinated trials.
Yes.
Means the variety cannot be released if it does not pass that AICRP trial, which is just the multi-locational trials. So from variety, it has gone into, from the release of variety from respective institutions, it has gone to AICRP and then it will be available for seed production.
Okay, ma'am.
Can you
know the difference between quantitative traits and qualitative traits? Smriti.
Hello.
Smriti,
sir?
Sir has asked the question. Can you please tell the difference between quantitative trait and qualitative trait?
Have you heard the term QTLs?
Have you heard the term QTLs?
Yes, ma'am.
What is the full form?
See, why I am asking is that you said you are going to use CRISPR technology for yield contributing traits. And yield contributing trait means, you are talking of quantitative genes. And there you cannot bring a small modification and bring the whole genome obeying that. Usually this technology is used where you have pinpointed that this is the gene and this modification will bring this difference. Okay, so since you are at a planning stage for your own work,
yes,
sir. Of a qualitative trait and not quantitative trait if you want to use genome editing.
Okay, sir.
Thank you. With this,
Just
we.
one small question.
Yeah, sure.
Any, Smriti, any reason why have you picked up this crop? Why have you selected this crop?
Actually, I want to work on legumes, so that's why I picked up chickpea.
Thank you.
Thank
want
you,
to
Smriti.
add one information for Smriti.
Yes, ma'am.
Yes,
Please.
ma'am.
Like what Dr. Khanuja has told, it is the qualitative trait which can be managed. Like I told you in rice also, they are looking for some abiotic stress gene, no? Like drought tolerant. And in case of chickpea, the work is already going on. This is for your information that nodulation, some nodulin related genes, already work is there where it is being targeted that whether it will have more nodulation or less nodulation.
Okay.
So that information is, that work is already going on in your Kanpur only, IIPR only. If
Yeah.
you're from, she's from Kanpur only, no?
She's from Kanpur. She has done 1 year of work in IIPR itself. And she had taken up this project. There was a literature cited that this particular gene has been seen and researched in soya bean. And they found, soya bean. Why don't you say about that? Why did you pick up this gene? Because the initial research by another group was done in soya bean, and we were looking into the effect of that gene in chickpea. So her study is focusing on that area.
Correct. So she can read about all these things.
Yeah, sure. Thank you for the inputs, ma'am. And with this, we end up the category of research. We have completed 7 presentations. Can we break for lunch or should we continue as the jury member suggests?
Yeah, lunch, I think. Dr. So what Dr. Srivastava says, Dr. Ashwini Srivastava or Dr. Rena.
There's a pending presentation from Avantika. She was facing issue.
Yeah, but I'm not able to connect with her, sir. Maybe if she's ready, we can take at the last after the 2 categories, that is innovation and entrepreneurship. I'll talk to her during lunch and maybe we can take up later on if she's ready, or else we'll drop it. So can we
can go for another half an hour.
Yeah, sure. We just get together at 2:15. Yeah. Thank you. So we'll take a break now.
So please reassemble at 2:15.
Yes, sir.
Thank you.
Dr. Soni, we'll stop the recording also so that we can bifurcate the recording.
Sure, ma'am.
And we can again.
Yeah.